Now everybody can see this. So very good morning to all the participants. I welcome you to the DHSC Online Academic Support Program. And today in this class, we are going to have a detailed analysis about the topic cell, the unit of life. And in this very chapter, we are going to conclude it by studying the cytoskeleton, the cilia, the flagella, and the centrioles in the very first class. And in the second class, we are going to have a discussion on the topic nucleus, chromosomes, and the microbiota in the family. Done? Okay. So without wasting any more time, so let's quickly jump to our part that is called as the cell as the unit of life. ये हमें start करेगा. Is it clear? समझते? I request every participants to kindly join and share your views. जहाँ भी जहाँ doubt रहे पिलामान तुम directly मते पचारी परिबा इठी class रे. You can chat, you can raise your voice. Okay. Now. So what is a cell? A very basic feature. What is a cell? What do you mean by cell? The cell is the basic fundamental, structural and functional unit of all living organisms. You know it very well. जब हम cell बोली होते हैं, cell की कौन पिला मन? Cell is a basic structural, basic functional as well as fundamental unit of life. You all know this about very beginning. हम जानते हैं जिनसे yes or no? तो cell हमको ଗୋଟେ fundamental structure provide करूची, हमको सेफ बॉडी रो दूची और तासु मेजर फंक्शन भी हम बॉडी रो करते जेम हम मेटाबॉलिक प्रोसेस हे गला एनाबॉलिक प्रोसेस कैटाबॉलिक प्रोसेस ईच एंड एवरीथिंग ऑफ ऑल लिविंग ऑर्गेनिज्म जति को लिविंग ऑर्गेनिज्म देखो छ लाइक ऑल दिस लिविंग ऑर्गेनिज्म प्रेजेंट ओवर हियर दे ऑल आर मेड अप ऑफ सेल्स ठीक है अब एक कथा मो कहूनी एक कथा सेल थ्योरी कहि जाइतले हमको कि एवरी लिविंग ऑर्गेनिज्म्स आर मेड अप ऑफ सेल्स एंड प्रोडक्ट ऑफ सेल्स यस और नो जतिक लिविंग ऑर्गेनिज्म देखो पिला मन दे ऑल आर मेड अप ऑफ सेल्स एंड प्रोडक्ट ऑफ सेल्स एंड देन असले साइंटिस्ट रुडोल्फ विल्को जे कहितले कि ओमने सेलुला ई सेलुला जार मीनिंग थिला कि सब जाक सेल्स माने अरेंज करछन तांकर प्री एग्जिस्टिंग सेल्स दिस वन तो दीज आर द थिंग्स हम एटा बहुत बिगिनिंग रो पढितले बहुत बेसिक जिंस हम एटा पढितले तो से बेसिक्स को हम एटी एंड करिके आगो जो नेक्स्ट. So you have seen those persons, you have known these persons. One is Robert Hooke, who first discovered a cell, and then it is Anton von Leeuwenhoek. He is the man who first discovered a living cell. The first our cell discovery is 1665 रे. Then our living cell discovery है our living cell discover करले के Anton von Leeuwenhoek. You all know about these persons. Now, a cell development of basic जो क्रेडिट जाउची काहाकू दिस माइक्रोस्कोप को तो जब माइक्रोस्कोप इन्वेंट केला सेबे जाके हम सेल को देखि पाइले तो विदाउट अ माइक्रोस्कोप वी कैन नॉट सी अ सेल इन अ नेकेड आई ठीक अछि हम नेकेड आई हम सेल को देखि परिबा नै सो दैट वी नीड अ माइक्रोस्कोप बिना माइक्रोस्कोप पे देखि हबो नै तो इन्वेंशन ऑफ दिस कंपाउंड एंड इलेक्ट्रॉन माइक्रोस्कोप्स दे रिवील द स्ट्रक्चरल डिटेल्स ऑफ द सेल. Now again, cells are basically two types. One is the prokaryotic cell, second is the eukaryotic cell. In the prokaryotic cell, you know the cells are divided into two categories on the basis of presence or absence of membrane-bound cell organelles. Those who are prokaryotic cell, they do not have any kind of membrane-bound cell organelles. In their body, there is no nucleus, no mitochondria, no Golgi body, no lysosome, no vacuole. nothing is there. But in case of eukaryotes, we will definitely find some membrane bound cell organelles. Then the pro unit is basically different cell, and our biological classification chapter re I already part ko पढ़ी सारिछे कि on the basis of the type of cell arrangement, on the basis of types of number of cells present in an organism, we have divided the entire biological classification, entire organism, living organism into 5 different kingdoms like the Monera, the Protista, the Fungi, the Plantae, and the Animalia. In the Monera part, you have included all the unicellular and prokaryotic cells. And then in the eukaryotic, then in the other 4 parts, we have concluded the multicellular things, the multicellular, the eukaryotic organism. So why we are discussing about this prokaryotic cell and eukaryotic cell? Because other topic जो अच्छे हमारे about the cilia, the flagella, then about the cytoskeleton से part जो हमें देखिएगा तो किसी part prokaryote माना पाखे थे खाने. Then as you can see over here, the long tail-like structure present here is called as a flagellum. So it is a bacteria. Can you tell me which kind of bacteria it is? By just looking at the shape of the bacteria, can anybody tell me which kind of bacteria it is? Dear participants, by looking at the type of cell, this picture, who can tell me which kind of bacteria it is? Any one of you? Well, the answer is these are lactobacillus. These are lactobacillus because they are rod-shaped structure. Even a rod-shaped structuration, that's why it's called as bacillus. Our lactic acid formation currently, milk to curd convert come, that's why it's called as lactobacillus. So basically the rod-shaped shape achivoli, these are called as bacillus. Yes. Now, तो हमें prokaryotic, eukaryotic, article details के basics देखेंगे हमें आगे जिबा. Eukaryotic cell, as you can see over here, in this eukaryotic cell, you'll find many cell organelles and many membrane bound cell organelles. और ता भीतर जो head अछि, the head of the department. Today we are going to discuss about the head of the department, the brain of the cell, that is called as the nucleus in the second class. Now you have a very basic idea about that. They have well organized membrane bound nucleus and membrane bound cell organelles. Then the presence of membranes gives a clear compartmentalization of the cytoplasm. Cytoplasm भीतर में बहुत सारी जगह हमें distribute कर दो से compartment formation कर दीजिए. They have a complex locomotory and cytoskeletal structure. That is the very important point of today's class. We are going to discuss about the same. That is a eukaryotic cells when achanti तांको पाखरे कौन अच्छी. They have a very complex locomotory and cytoskeletal structure. So what do you mean by these two words, locomotory and cytoskeletal? See, locomotory word has been derived from locomotion, and locomotion is nothing but the voluntary movement of a person or something from one place to another place. Right now, I'm doing locomotion from point A to point B. I'm just doing locomotion. So voluntary movement होची गोटे जगह और जगह को movement होची. तो सेल को movement करिया को हेले, the organism को movement करिया को हेले, the organs को movement करिया को हेले, they need some locomotory organs. और से locomotor structure किए provide करूची ଆମର ସାଇଟୋସ୍କେଲେଟନ ପ୍ରୋଭାଇଡ କରୁଛି ତାକୁ ସେ ଜିନିଷକୁ ଯେମିତି ଆମର ସିଲିଆ ଫ୍ଲାଜେଲା ଆଉ ସେମାନେ କାହାକୁ ନେଇ ତିଆରି ହେଇଛନ୍ତି ସେ ସାଇଟୋସ୍କେଲେଟନ ଲାଇକ ଦ ମାଇକ୍ରୋଟ୍ୟୁବୁଲ୍ସ ଲାଇକ ଦ ଆମର ଟ୍ୟୁବୁଲିନ ପ୍ରୋଟିନ କୁ ନେଇ ତିଆରି ହେଇଛନ୍ତି ମାଇକ୍ରୋଟ୍ୟୁବୁଲ ଇଣ୍ଟରମିଡିଏଟ ଫିଲାମେଣ୍ଟସ ହେଇଗଲା ମାଇକ୍ରୋଫିଲାମେଣ୍ଟସ ହେଇଗଲା ସେ ବିଷୟରେ ଆମେ ଆଜି ପଢିବା clear. And then in the last part, we are going to study about the genetic material is organized into the chromosome. Cell, because we have genetic material, so code storage inside the chromosome. And we all know these chromosomes are the vehicle of heredity. Chromosomes they are the vehicle of heredity. So, we transfer our gene of interest from one person to another person. Yes? So, these are the very basic things. And dear participants, हम पाखरे जो सेल थिला प्लांट सेल हो बा एनिमल सेल हो ता पाखरे हम 11 टा पार्ट डिवाइड करथिले हम एनसीईआरटी सिलेबस ता 11 टा पार्ट डिवाइड करथिले एंड each year एटु क्वेश्चन आसी थे पिला माने ସେଥିପାଇଁ କି ଏଇ ଜିନିଷ ଉପରେ ଟିକେ ଫୋକସ୍ କରି ବେସିକାଲି ରଖିଥିବ। ଦେନ୍ ଯେମିତି କି ହେଇଗଲା ତୁମର ସେଲ୍ ମେମ୍ବ୍ରେନ୍, ସେଲ୍ ୱାଲ୍, ଏଣ୍ଡୋମେମ୍ବ୍ରେନ୍ ସିଷ୍ଟମ୍, ଚାରିଟା ପାର୍ଟ ଆସିବ ଏଣ୍ଡୋପ୍ଲାଜମିକ୍ ରେଟିକ୍ୟୁଲମ୍, Golgi apparatus, lysosomes, vacuoles, then mitochondria, then plastids, then ribosomes. So we have completed till this. Yes or no? We have completed till this. Some of the participants have not muted themselves, so it's a kind request to everyone. Those who have not muted, kindly. Okay. See, so we have concluded till this. We have completed about the cell membrane, the cell wall, the endomembrane system. I myself completed this part. Then the mitochondria, then the plastids, then the ribosomes. Today in this very class, we are going to discuss about the cytoskeleton. We are going to discuss about the cilia and the flagellum. Then we are going to discuss about the centrosome and centrioles, and then we are going to discuss about the nucleus, and in the very last part, we are going to have a quick revision about the microbodies. So in today's class, these are the scenarios, these are the parts. I'm a menu rahila ita, I'm a part of plan start with a done. Okay. The cell membranes, cell wall, endometrium system. Some of you have not muted till now. No. Okay. So starting from the very basics, that is cytoskeleton. What is the cytoskeleton? First thing first, what do you mean by cytoskeleton? And the answer lies in the name. आंसर आम देखियो साइटोस्केलेटन आंसर ना रे ही लुचैकी अछि साइटो प्लस स्केलेटन साइटो you all know cyto means cell so no in biology we have already discussed cytology that is the branch of biology which deals with the study of cells is called as cytology तो cyto माने हेगला cell तो माइटोसिस मेयोसिस के भी देखि जे जिंस पिला माने कि सेल डिवीजन जो होची हम ताको साइटोकाइनेसिस बोली कहूची तो साइटो माने हेगला सेल काइनेसिस माने डिवीजन थिला the cytosol means cell और जो skeleton part अछि it's a framework ଆମ body ର ଗୋଟେ framework ଯେମିତି ଆମ body ରେ skeleton ମାନେ ଅଛନ୍ତି body ଗୋଟେ shape ଦେଇଛନ୍ତି body ର shape କୁ maintain କରୁଛନ୍ତି just like that cell ଭିତରେ ଯୋଉ cytoplasm ଅଛି ଆଉ cytoplasm ରେ ଯୋଉ embedded ହେଇକି ଅଛନ୍ତି different cell organelles ମାନେ like the endoplasmic reticulum the Golgi apparatus the lysosomes the vacuoles and the mitochondria, the chloroplast, and all those cell organelles who are present inside the cytoplasm. So they need a framework. जहाँ ऊपर समान सेट नहीं करेंगे. तो से framework basically क्या provide करते cytoskeleton. If we go to the definition of cytoskeleton, what does it mean? That it is a network of filamentous proteinaceous structure present in the cytoplasm. So single line by NCERT line जब हमें देखिएगा ये कहाँ को नहीं कह रही थी? It's a filamentous. Filamentous means branching. ये branch है जो network से बाहर होता है. It's a filamentous and a very important point proteinaceous. Yes, you have heard right. Proteinaceous. कि ये जो गोटे single word ଅଛି ना, ये चुने ही question ଆସିବ. When I say proteinaceous, that means it is made up of proteins. It is basically made up of proteins. What are proteins? In the biomolecular segment, we have already discussed what is a protein. Basically, protein is a polymers. Protein is a polymer, a bunch of amino acids joined together through peptide bond to make a polypeptide chain that is called as protein. So, say the protein is a group of amino acids mane mistile. Different amino acids mane mistile. Heteropolymer of amino acid, you can call it as a protein. तो ये जो प्रोटीन अच्छी से प्रोटीन को नेकी तैयारी हिचरी को मैंने that the cytoskeleton तैयारी हिचरी ଆଉ ଏଇଟା ଗୋଟେ କଲେକ୍ଟିଭଲି ନେଟୱାର୍କ ସିଷ୍ଟମ ଅଛି ସେ କଲେକ୍ଟିଭ ନେଟୱାର୍କ ସିଷ୍ଟମରେ କିଏ କିଏ ଆସିବ ନା ମାଇକ୍ରୋ ଟ୍ୟୁବୁଲ୍ସ ଆସିବ ମାଇକ୍ରୋ ଫିଲାମେଣ୍ଟ ଆସିବ ଆଣ୍ଡ ଇଣ୍ଟରମିଡିଏଟ ଫିଲାମେଣ୍ଟ ୟେସ୍ ଆମେ ତିନିଟା ପାର୍ଟ ଡିଟେଲ୍ସରେ ପଢିବା ଇନ ଦ ସେଗମେଣ୍ଟ କଲ୍ଡ ସାଇଟୋସ୍କେଲେଟନ I hope तुम cytoskeleton और basic definition पाई गलनी. What is a cytoskeleton? को भी टॉपिक को बुझिया पाई कि I always prefer कि go and break down the topic into 5 different parts, and that 5 different part is the basic questions: what, when, where, why, and how. So what is cytoskeleton? The cytoskeleton is a filamentous proteinaceous network present in the cytoplasm. ठीक अछि और filamentous proteinaceous network टी अछि जोटी के हमर cytoplasm में present अछि और तार काम कोन कोठी अछि where it is present inside the cytoplasm. Okay, inside the cytoplasm of which cell? Prokaryotic cell or eukaryotic cell? And the answer comes here, it is the eukaryotic cell. The cytoskeleton is not present in case of prokaryotic cell. They are present only in eukaryotic cell because prokaryotic cell organelle ही नांती जो मानक भी होल्ड करके रखियो जब eukaryotic मानक पाखे बहुत सारा organelles अछंती बहुत नंबर organelles अछंती ସେଥିପାଇଁ कि they are present in the eukaryotic cell. And what is the function आपको role कौन why they are here काम कौन main काम है तीनटा एम को नहीं बुझिबा 3M को नहीं काम हेची आ से 3M हे गला के पिला माने what are the 3M? First M is your provides the mechanical support. इधर गोटे mechanical support provide करिवो providing mechanical support, and then providing M रे motility, and then M रे maintenance of the shape of the cell. काम करे साइटोस्केलेटन जो अछि आर तीनटा काम रहिला फर्स्ट आसिला एम रे एम सेकंड रे आसिला एम एंड थर्ड रे भी आसिला एम ठीक अछि फर्स्ट एम रो काम हेला कोन एठी मोटिलिटी तो सेल को ये अबिका मोटाइल करिवारे हेल्प करछि ऑर्गेनेल्स मानन को अबिका मूवमेंट करिवारे मोबाइल करिवारे हेल्प करछि मोटिलिटी ऑफ द सेल सेकेंड आउ कोन करछि अबको सेल को कोन करछि ना मैकेनिकल सपोर्ट दोछि it provides mechanical support to the cell mechanical support to the cell and this provide करछि and the last या कोन करछि maintenance of the shape of the cell maintenance of the shape of the cell. Now, these are the three basic words has been done by this cytoskeleton. It's a network of filamentous proteinaceous structure present in the cytoplasm and our main काम हेगला cell को motility provide करिबा mechanical support दबा ता सहित maintenance provide बुझ पाइला dear participants any doubt till this okay. So we are going to see a very clear picture about and get a clear idea about the cytoskeleton and I will convergence search. You can see upper part रहा cell membrane और plasma membrane, तले भी रहा cell membrane और plasma membrane. In between जो part रहा से part रे हमर कोमन अछि there is present are what all the cell organelles and the cytoplasm and of course the cytoskeleton. So this is a membrane. The yellow color sheet you are looking over here, that is a cell membrane. The last yellow color membrane, that is also a cell membrane. In between the two cell membrane, what is there? There is embedded all the cell organelles, the cytoplasm, and is सब जिनको कहाँ ऊपर डिस्ट्रिक्ट है कि ताको टिके के रखी थी. The answer is your cytoskeleton. ये cytoskeleton तो मैं देख पा रहा हूँ तीन प्रकार पार्ट रे हम लोग डिवाइड करछे. One is your one is your microtubule for tubulin. Number one is your microtubules and tubulin तैयारी करने मन so microtubule is a structure जहाँ कहाँ को नहीं बनी को प्रोटीन को नहीं की बनी ट्यूबुलिन प्रोटीन को नहीं बनी then you are having intermediate filaments ये फोन कहाँ को नहीं बन चुके कहीं पर आगू जाएंगे and then microfilaments they are made up of this actin प्रोटीन को नहीं तैयारी करते ठीक है ये lamin तापरे भाइमेंटिन then keratin ये सब प्रोटीन को नहीं मन तैयारी करते these all these three things are proteinaceous in nature they are made up of protein even कहाँ को नहीं तैयारी करते प्रोटीन को नहीं की तैयारी करते now our structure and नारी लिखाई जी microtubules तो ट्यूब रो जो भीतर होलो पार्ट अच्छी that is called as tubules. और ये ट्यूबुल जो रहिला they have two components. You can see one is light green another is dark green. तो लाइट ग्रीन जदि अल्फा कंपोनेंट हैला अल्फा ट्यूबुलिन कहिबा, देन जो डार्क ग्रीन कम कहिबा बीटा ट्यूबुलिन. एंड एमाने अल्फा ट्यूबुलिन गोटे स्ट्रक्चर बनेबे, देन बीटा ट्यूबुलिन स्ट्रक्चर बनेबे. तो से स्ट्रक्चर ना हैला प्रोटोफिलामेंट्स. तो जो अल्फा ट्यूबुलिन बीटा ट्यूबुलिन एमान जो प्रोटोफिलामेंट फॉर्मेशन करिए पिला माने एही आगो जाइके कोन फॉर्मेशन करिवो ना माइक्रोट्यूबुल नेटवर्क के फॉर्मेशन करिवो. मन रखिता हमर जो तीनटा साइटोस्केलेटन माने अछंती तो जदि हम तार साइज देखिबा देन माइक्रोट्यूबुल इज द लार्जर वन, सबुटु बड़, as compared to intermediate filament. As the name suggests, intermediate is in between, and सबुठ सानरे की आसी तुम्हारा microfilaments. Yes or no? The microtubule is larger than your intermediate filaments, which is larger than your microfilaments. ठीक है. आउ ए जो तीनटा जिंस हम कहली आंकर डिटेल्स फंक्शन कोन है, माने को जिंस रे हेल्प करतन ताको हम डिटेल्स से बारे बुझिबा. फर्स्ट ऑफ ऑल, you just see the structure. This green color part जो तुम शो कर that is the microtubule. Then ये जो blue color part तुम देखो जो पिलामन, this blue color part is called as the intermediate filaments, and सबुठ last part जो भी purple color parts of version that are called as your microfilament. Yeah. Yes, ये जंके I hope तुम सब बुझ पारछो ये जिंसटी. Then basically जानी पारे एठी you can see a picture of a cell. हम ताको इलेक्ट्रॉन माइक्रोस्कोप रे स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप रे पिक्चर सब आसी पिला माने ए पिक्चर हम देखि जानी पड़छे कि it has column of tubulant dimers. एमन बहुत सारे डाइमर्स माने मिसी के tubulant डाइमर. बट ये जो डाइमर बेसिकली कहा को नेते दीटा मोनोमर को नेके तयारी छि. दीटा मोनोमर, वन इज जो अल्फा मोनोमर, वन इज जो बीटा मोनोमर. आ ये अल्फा देन बीटा ए माने मिसी के बनैछन कोन गोटे ट्यूबुलिन डाइमर बनैछन. एंड ए जो डाइमर माने अछन अल्फा, ये रहला अल्फा, and it comes the man. तो अल्फा बीटा मिसी के हमारा स्ट्रक्चर बनैछन that is called as tubulant dimer. ओके. जे तुम कहितले कि माइक्रोट्यूबुल्स इज greater than intermediate filaments, and then microfilament is border रहिबो. Intermediate filament is greater than your microfilaments. Now ए जो माइक्रोट्यूबुल अछि, माइक्रोट्यूबुल रो डायमेंशन केते देछि, लेंथ केते देछि 25 नैनोमीटर. पिला मन भलसे मन रखियो जेहेतु ए माने गोटे सर्कल टे बनैछंदी आ आंका पाखरे बहुत सारा माइक्रोट्यूबुल्स माने एमते डायमंड बनै के अछंदी. तो आ जदि बाहर ल्यूमेन को मु देखिबी, आउटर साइड को देखिबी, ताहाली तार डायमीटर केते हे गेला, दिस आउटर साइड डायमीटर केते हे गेला 25 नैनोमीटर. बट इफ आई गो एंड लुक एट द इनर लुमेन, the lumen diameter में देखिबी, then inner part length हे गेला 15 नैनोमीटर. क्यों आमे सेत पई कि बाहर पार्ट को स्टैंडर्ड हिसाब रे मापी थाउ और बाहर को 25 नैनोमीटर हे कि ही नहीं थोई. If we come to the second वाला part, second के गेला the fibrous subunit that is called as keratin ने तयारी हितियो जिंस, that is your intermediate filament. अंकर जो डायमेंशन तमे देखियो पिला माने it is more or less 8 to 12 नैनोमीटर जाई थांती. Then and the सबुट smallest जो part अछि that is the actin subunit, and this actin subunit को नै कि तयारी छि माइक्रोफिलामेंट. आको actin filament भी कहे जे. आर बेसिकली तुमे काम देखो कोठी मसल फाइबर मानन पकरे जे मसल कंट्रैक्शन करि थाए, लोकोमोशन एंड मूवमेंट चैप्टर तुम्हें पढ़िबो धीरे धीरे. इट जो मसल माने हमर कंट्रैक्शन करथाने देन सेई टाइम रे हमर एक्टिन सब यूनिट्स माने काम रे आस्थांती. एंड आंकर साइज सब छोटो दैट इज जस्ट 7 नैनोमीटर. दिस इज 25 नैनोमीटर. आउटर साइड जो देखिबी, इनर साइड देखिबी 15 नैनोमीटर रे लेंथ रहबे माने. देन तापर 8 टू 12 जेंग रहला इंटरमीडिएट फिलामेंट, एंड देन सबुट छोटो रे के पळे आसियो माइक्रोफिलामेंट 7 नैनोमीटर स्ट्रक्चर रे के दैछि. Okay, the participants, some of you have not muted till now. I am requesting everyone. Now, is it clear till this? What is your my cytoskeleton na कौन part अछि cytoskeleton तीनटा जिनस हम कौन जानले is it clear till now any doubt you wanna ask the participants any doubt you wanna ask from this segment basically your microtubules your microfilaments and your intermediate filaments anything you need to any query you have till this okay. If you have any doubt you can ask me in the chat box otherwise you can unmute yourself and ask me the questions okay so you can see in your screen about the slide that is the cytoskeletal filament and filament आगे तीनटा जिनसे हम लोग divide करते the three types of filaments found in the cytoskeleton are they are distinguished by their size, their structure, and the protein subunit of which they are made of. So, केमिकल size रे अछंदी, the structure केमते अछि, आ कोन protein ने समान तेरियन से बेसिकली हम ताको डिफिकली divide करछे. If you will go and look into the microfilament, the actin filament को तमे देखियो filament एटे microfilament को देखिया start करिवो. These microfilament are strands in double helix structure. What is double helix structure? This part is called as double. This is a one helical structure and this is a second by double helical structure. A bada presence man just like your DNA. One side is positive end and one side is negative end. What is this positive and what is this negative end? Positive or negative mane kono? I have already told you that these things that have some produce pila mane they are your proteinous structure and protein means kahaku nahi tiyari che bunch of amino acid ko. So whenever you see an amino acid the alpha carbon has four parts. One side it is COOH and second side it is NH2 and जब एमाने आपस में बॉन्ड बनने की ଗୋଟେ ନୂଆ प्रोटीन टे बनेबे this is your H and this is your R1 then again your R2 and this is your H ଯେତେବେଳେ ବି ଆଙ୍କ ଭିତରେ ବଣ୍ଡ ଫର୍ମେସନ ହବ ପେପ୍ଟାଇଡ ବଣ୍ଡ ଟେ ଫର୍ମେସନ ହବ ଆଉ ୱାଟର ମଲିକ୍ୟୁଲ ଏଠି କଣ ହେଇଯିବ ରିମୁଭ ହେଇଯିବ ଦେନ୍ ତମେ ଦେଖିପାରିବ ଗୋଟେ ପଟେ ତାର ଏସିଡିକ ଗ୍ରୁପ ରହିବ ଗୋଟେ ପଟେ ଆମିନ ଗ୍ରୁପ ରହିବ ଏସିଡିକ ଗ୍ରୁପ ଟା ଆଗକୁ ଯାଇକି H କୁ କାଟିକି ଏଠି ମାଇନସ ହେଇଗଲେ and ଏପଟେ NH3 ପ୍ଲସ ନ୍ୟୁଟ୍ରାଲ ଏଣ୍ଡ କଣ୍ଡିସନ କୁ ପଳେଇ ଆସିବ ତ ଗୋଟେ ତାର ନେଗେଟିଭ ଏଣ୍ଡ ରହିଲା ଗୋଟେ ତାପରେ ପଜିଟିଭ ଏଣ୍ଡ ରହିବ ଦ୍ୟାଟ ଇଜ ଦ ସିନାରିଓ Next, there's a double helix strand cell ये actin filament सबुठु छोटो filament actin filament actin territory. Okay, what is the function of this filament? ना ये जो microfilament वाला function कौन? It maintain the cell shape. ये सेल द शेप को मेंटेन करछनी बाय रेजिस्टिंग द टेंशन ये टेंशन माने हमर नॉर्मल बायोलॉजिकल टेंशन कथा पड़े बे टेंशन पहले हमें कहा कथा बुझी टेंशन कह जाई फिजिक्स में टेंशन कथा आसिला जो टेंशन माने गोटे मॉलिक्यूल तापर मॉलिक्यूल टे केमते बाइंड हे होजी ताको जरा रस्सीटा दोरे आउरी गुडे हे जो हेड जो टेंशन क्रिएट हबो से टेंशन माने रिलैक्स करे जानती रेजिस्टिंग द टेंशन then it moves cells via the muscle contraction or cell crawling जब सेल अबिका मसल कंट्रैक्ट करछि बट को जिंस उठौछि तो डंबल टे उठेले तो मसल टे कंट्रैक्ट करला फोन टे छाडेले से रिलैक्स होला तो कंट्रैक्शन रिलैक्सेशन ए टाइम पर कोन होबो ना हमर जो सेल माने मूव करछन से सेल को मूव करे हेल्प करछि कि ना हमर एक्टिन फिलामेंट और माइक्रो फिलामेंट हेल्प करछि देन आकर आउ गोटे मेजर रोल कोट पाइबो तुमे ना इट हेल्प इन डिवाइड एनिमल सेल्स इनटू टू तो गोटे सेल टे डिवाइड करछि वाला सो सेल फ्यूरा फिलामेंट जो फॉर्मेशन होछि दिस इज एन एनिमल सेल इट हैज टू and then so did a nucleus to animal cell टी आगु जाई डिवाइड हबो गोटे सेल फ्यूरा फॉर्मेशन करिवो और जो सेल फ्यूरा फॉर्मेशन कला बेले एई बडा जो स्ट्रक्चर बनिवो ए स्ट्रक्चर बनिया पई कि ना हमर कहार हेल्प हम नोचे एक्टिन फिलामेंट र हेल्प नोचे and then ये स्ट्रक्चर टी आगु जाई कन्वर्ट हेजिवो टू डिफरेंट सेल that is called as mitosis and last it move organelles and the cytoplasm in case of plants in case of fungi and in case of animals as you can see kingdom plantae kingdom fungi kingdom animalia all those three things belongs to what eukaryotic cell so सब eukaryotic cell मानो पाखरे they all are eukaryotic multicellular organisms. They all are eukaryotic multicellular organisms ମୋ ପାଖରେ କଣ ଅଛି ନା ସାଇଟୋପ୍ଲାଜମ ଅଛି ଆଉ ସାଇଟୋପ୍ଲାଜମ ରେ କିଏ ଅଛି ସାଇଟୋସ୍କେଲେଟନ ଅଛି ଆଉ ସାଇଟୋସ୍କେଲେଟନ ର ଗୋଟେ ଟାଇପ ହେଇଗଲା କିଏ ସାଇଟୋସ୍କେଲେଟନ ଗୋଟେ ଟାଇପ ହେଇଗଲା କିଏ ତୁମର ନା ମାଇକ୍ରୋଫିଲାମେଣ୍ଟ ଆଉ ଏଇ ଯୋଉ ମାଇକ୍ରୋଫିଲାମେଣ୍ଟ ରହିଲା ସେ ମାଇକ୍ରୋଫିଲାମେଣ୍ଟ ଏଇ କାମରେ ସବୁ ହେଲ୍ପ କରୁଛି ଦେନ୍ ତ ଗୋଟେ ବେସିକ ଆଇଡିଆ ପାଇଗଲ what is microfilament what is microfilament what is actin filament ଆଉ ସେ ଯୋଉ actin filament ବା microfilament ର structure କଣ ତାର size କେତେ ଅଛି then ସେ କୋଉ form ରେ ଥାଏ କୋଉ ଜାଗାରେ ଥାଏ and what is the function the main important thing is function ଏଠୁ question ନିହାତି ଆସିବ ଆଉ ମନେ ରଖିବ diameter 7 nanometer and ସବୁଠୁ smallest ଥାନ୍ତି ଆଉ କାମ ହେଲା maintain the cell shape of the body move cells by muscle contraction or cell crawling divide animal cells into two animal cell ମନେ ରଖିଥିବ plant cell କଥା କହିନି animal cell କଥା କହିଛି ତ animal cells ଏମାନେ କଣ କରୁଛନ୍ତି cell furon ଯେତେବେଳେ cell divide କରୁଛି ଯେବେ ସାଇଟୋକାଇନେସିସ୍ ହଉଛି ଆଉ ଏଠି ସାଇଟୋକାଇନେସିସ୍ ରେ cell furo method କଥା ଆସିବ ତ ସେ cell furo formation କରିବାରେ ଏମାନେ help କରିଥାନ୍ତି ଆଉ then they divide the animal cells into two and move organelles and cytoplasm in plants in case of fungi and in case of animals now comes the number two that is the intermediate filament why it is called as intermediate its size comes in between the microfilament and the microtubules microtubule being the larger one and microfilament being the smaller one in between there is a segment that is called as intermediate filaments and these fibers wound into thicker cables एमाने आहुरी थिक केबल को बा थिकर केबल को अबिका wound ହେଇଛନ୍ତି ବା rounded ହେଇଛନ୍ତି ଯେମିତି କି ଆମ ସାଇଜ ତମ ପାଇବ ଅବିକା somehow from 8 to 12 nanometer ତାକୁ ଆମେ 10 nanometer ନେଇକି କେସ ଦେଖୁଛୁ ତ 8 to 12 nanometer size ମାନ ଥାନ୍ତି they are made up of keratin they are made up of hymentin they are made up of lamine or other protein what is keratin you all know keratin protein is present in our hair our hair cell man present in keratin proteins ମାନେ Vimentin is kind of a protein. Lamine is a kind of protein. Lamine code सिर्फ नहीं जो न्यूक्लियर मेम्ब्रेन जो सेल मेम्ब्रेन से मेम्ब्रेन ना भीतर पर ଗୋଟେ ଲାମିନା ଟି ଥାଏ ସେ ଲାମିନା କୁ ବନେଇଥାନ୍ତି କି ଲାମିନ ପ୍ରୋଟିନ the lamine protein formation କରୁଛି we call them as lamina here the lamine protein ने A1 ଜିନିଷ ସବୁ ମିଶିକି ଆକୁ ତିଆରି କରିଛନ୍ତି ଆଉ main କାମ ହେଲା it maintain the cell shape by resisting tension just like the microfilament and then the most important part it anchor nucleus and some other organelles ଆମର the brain of the cell ଅଛି ତାକୁ ଆମେ support provide କରୁଛନ୍ତି anchorage provide କରୁଛନ୍ତି mechanical support provide କରୁଛନ୍ତି and which is very crucial you all know in case of plant cells the vacuole can take upon like the 80 to 90% of the entire cell part सेल जो स्पेस अछि 90% केवल vacuole ही नहीं जीव तो आउ जो रेस्ट 10% एरिया रहला तार भीतर सब जिंस को हमको होल्ड करे अछि तो इन दैट वेरी पर्टिकुलर सेगमेंट हमको एटी माइक्रोफिलामेंट माने इंटरमीडिएट फिलामेंट माने हेल्प करता है सेल को ठीक से होल्ड करिबारे एंकरेज का सपोर्ट प्रोवाइड करिबारे ओके आई गॉट सम 3 chats very good morning Banmali any doubt you can directly text me in the chat box now the last part asila ka kotha eti microtubules katha and the name रे ही ना लिखा ना भितर जिंस अछि tubules हेगला the hollow tube तो hollow tube रे प्रेजेंट अछंदी and आर जो लेंथ हेगला 25 nanometer जो तुम देखियो बाहर पटटा और जो भीतर पट देखियो तो lumen भीतर पार्ट देखियो then it is called as 15 nanometer साइज रहबो then आंको पाखे दी प्रकार डायमंड अछंदी गोटे अल्फा आ गोटे बीटा ट्यूबुलिन and एमाने आपस में मिसिकी बनैछंदी कोन गोटे प्रोटीन डायमर बनैछंदी और डायमर माने कॉइल हे कि बनैछंदी माइक्रोट्यूबुल आंकर मेन काम कोन हेला ना इट मेंटेन द सेल शेप by resisting the compression that is the pushing रे हेल्प करंती then it moves cells via flagella और cilia इस सेल को जो flagella अछि जो cilia अछि its flagella cilia all are the locomotive organs जो माने मूवमेंट रे हेल्प करथन सेल को तो से माने सब मूव करिबारे माने हेल्प करथांती then it move chromosomes during the cell division you very important part the chromosome rahila amara the hereditary material the vehicle of heredity this chromosome needs to be separated and এটা কো টাইমে বেসিক্যালি হেইতা during the cell division जब हम cell divide করছে when we are dividing the single cell into two or four depending upon the type of division, that is mitosis or meiosis. This cell divide kala bale the chromosomes should get equally divided and go to two different parts. बुझल आ ए क्रोमोसोम्स माने को सेपरेट करिया पाई कि तांको दी जनो दीटा सिस्टर क्रोमैटिड को आपस में अलग करिया पाई कि we are getting help from the spindle fibers. और जो spindle fiber जो formation करते हैं they are nothing but are microtubules. जो microtubules माने actually microfilaments माने actually by intermediate filament माने actually they all helping to do so all these parts. They assist in the formation of cell plate during the plant cell division. मतलब कि बस cell जो division in case of animal help करथिले कि हमारा actin filament और microfilament, but in case of plant cell division को के help करछि जो cell plate method formation हबो तो के help करछि, the answer is your microtubule. Then it also moves larger organelles and it provides tracks for intracellular transport. पिला माने this part is very important आपको तो मन रखिबो. तो ये जो हमारा माइक्रोट्यूबुल रहेगा इट एक्ट लाइक द सेल ट्रैफिकिंग सेल सिग्नलिंग जब होता है सेल ट्रैफिकिंग जब होता है तो वहां कमला रोड बनेगा तो ये रास्ता दिखाई थे सेल कोड रोड बनो के साइड रोड बनेगा सिर्फ साइटोस्केलेटन ना मन से सेल सेल जंक्शन को मन कनेक्ट करते हैं तो ये मन आपन कार्गो प्रोटीन अच्छे से कार्गो प्रोटीन ना हेल्प रे मन सेल को व इंट्रा सेलुलर ट्रांसपोर्ट कम्युनिकेशन करे मन हेल्प करते हैं. So TK is right up also देखिए दियो. You can take a screenshot if you want to do so. And in case of any doubt, you can ask me in the chat box. The participants, if you have any doubt, you can ask me in the chat box. Yes, dear participants, any doubt till now? Shall we move ahead? आगो बढ़िबा. Any doubt अछि पचारियो चाहिबो? Dear participant, बनमणि, वर्षा, मानस, नीतू, प्रज्ञा, सीतामणि, सोनाली, स्वीटी, एने डाउट कि किछ पचारियो चाहिबे पिला माने ना हम आगु जिबा कंटिन्यू करबा ई पार्ट को. Okay. The next segment asila. Okay. I got a message. Okay, one more. Our second part I'm start regarding the cilia and about the flagella. See, so what is cilia? What is flagella? They are the tiny hair-like outgrowths of the cell membrane. You can hold the छोटे tiny hair-like outgrowths when present around the cell membrane. Cilia, the small structures which work like the oars and it causes the movement of the cell or surrounding fluid. बहुत से मुझे part था कौन कौन करती. ना मुझे cilia रहीगला पिला माने cilia हो बा flagella हो. They are tiny, tiny माने पिला माने हेगला small. They are tiny or small hair-like outgrowth. ठीक है hair like outgrowth गोटे part रे माने बाहर को एम सब hair-like outgrowth है कि भारती. I am talking about cilia. Then ये जो cilia are small structures which work like oars. ये जो oars वाला movement में कहूँ ये जो डोंगा जिंदा आहुला मरा हुए देखिए जब पिला में डोंगा रे बसले दी पटो आहुला मरा ये डोंगा को आगो पठावे जब पानी को पछू टाणुचे सो दैट हम डोंगाटी आगो जाई परिबो एग्जैक्टली से मूवमेंट हम ओर्स मूवमेंट बोली कहिथि और से ओर्स मूवमेंट कला बेलु एमान ताको जे टर्बुलेंस क्रिएट करन से डायग्राम तुम आगो देखिएबे ये जो पार्ट तुम सिलिया माने भाग चंदी ये माने यार्ड को बोलिले देन आउ किछि सिलिया अछने यार्ड को मूव करिवे सो अबिका एंगल ऑफ मोशन ए पटो रही परिबा मतलब दे एक्ट लाइक ओर्स. Next, it causes the movement of a cell or the surrounding fluid. ये दिड़ा जिनसे करता है या तो सेल रा मूवमेंट करी परिबे उते जगह रे कि सेल रे हलचल किछ कर परिबे और जले सेल स्टेबल रहले सराउंडिंग फ्लूइड रे किछ हलचल कर परिबे भलसे बुझे पार्ट को दे कैन डू मूवमेंट इन द सेल ने रही के मान सेल ने मूवमेंट करिवे अदरवाइज से माने बाहर जो एनवायरनमेंट अछि सेल से एनवायरनमेंट किसी गोटे हलचल स्टार्ट करि परिबे गोटे मूवमेंट क्रिएट करि परिबे एंड आई एम सेइंग मूवमेंट don't confuse this term with locomotion because there is a significant difference between movement and locomotion. It is बहुत important जिनसे भलसे सुने था all locomotions are movement but all movements are not locomotion. तो राइट ना मोर जगह छिड़ा ही कि मोटे मूवमेंट करि पारछि मु तुम कहू जे एटा एमती हबो हेटा सेमती हबो लाइक दिस लाइक दैट एंड ऑल दैट इज कॉल्ड एज मूवमेंट यस नो और तुम सेपटे स्क्रीन सेपटे बसिकी फोन रे देखला बेलू बुझला बेलू मुंड एमती मूवमेंट करि पारतो दैट इज कॉल्ड एज मूवमेंट बट लोकोमोशन कोन हे गेला लोकोमोशन इज वॉलंटरी मूवमेंट बा वॉलंटरी एक्शन फ्रॉम वन प्लेस टू अनदर प्लेस देन अब जते बेले मु गोटे प्लेस बा गोटे प्लेस को जाउची सेते मोर अपिका मूवमेंट होची हैंड मूवमेंट होची लेग मूवमेंट होची. So all locomotions are movement, but all movements are not locomotion. The confusion बनी cilia जो होची it helps in movement. बारंबार कहूची cilia रे काम होला कोन हेल्प करिबा movement रे हेल्प करिवो या तो से सेल ना movement रे हेल्प करिवो और सेल ना सराउंडिंग जो फ्लूइड अछि से फ्लूइड को अब movement से हेल्प करिवो. But flagella is longer as compared to cilia. ये cilia ठारू कौन थे बड़ा थी वो longer than the cilia and it responsible for the cell movement and flagella prokaryotic cell and eukaryotic cell are structurally different थांती. तो flagella जो बची पिला माने locomotion ने help करियो. flagella गोटे जगह और जगह जीवा ପାଇଁ हेल्प करिवो तार अपेक्षा जो मूवमेंट जो से करिवो से ओर्स लाइक मूवमेंट करिवोनी से तार डिफरेंट मूवमेंट करिके पेंडुलम टाइप रो मूवमेंट करि से आगु जीवा बड फ्लैजेला जो अछि और फ्लैजेला प्रोकैरियोट भी थाए फ्लैजेला यूकैरियोट भी थाए बट देयर इज स्ट्रक्चर डिफरेंट प्रोकैरियोट फ्लैजेला अलग तयारी होते हैं यूकैरियोट फ्लैजेला अलग तयारी होते हैं. तो ये गोटे सेल रो मेम्ब्रेन हे गला और ये सेल मेम्ब्रेन ट्रांजिशन फाइबर पाखरु बाहर गोटे आउटग्रोथ हे कि बाहर छी से आउटग्रोथ ना हे गला कोन सिलिया और फ्लैजेला. These are the basal body. This basal body otherwise called as blepharoplast और आउ गोटे ना हे गला blepharoplast और एट वे तारा जो देखियो द कोर पार्ट जो रहला दैट कोर पार्ट इज कॉल्ड एज axoneme. तो axoneme is the core part present in the cilium. Any doubt till this? You can see very well. Kindly tell me the name of the two organisms. लेमने होमवर्क के लिए अबिका म 2 मिनट रे आंसर दबो व्हाट आर द नेम ऑफ दिस टू ऑर्गेनिज्म दीटा ऑर्गेनिज्म ना कोन अछि बनमाली वर्षा मानस नीतू प्रज्ञा स्वीटी I am questioning all of you. Can you just tell me the name of these two organelles? I'll be waiting for your answer. Yes, any answer I got? No answer till this. पिलामना ना कोन? Who can tell this? केही जदि पार्टिसिपेट करबोनी then how can you expect कि हमें बुझ परियबोनी जिंसटी? It's very simple. This structure is called as your euglena. हमें पढ़ी से बायोलॉजिकल क्लासिफिकेशन चैप्टर रो जो हमर थिला किंगडम मुनेरा देन किंगडम प्रोटिस्टा देन किंगडम फंगी देन किंगडम प्लांटी एंड देन किंगडम एनिमलिया ये पांचटा किंगडम रो जब किंगडम प्रोटिस्टा पढ़थिले सेतला मैं आंसर टी पाइथिले ना यूग्लिना प्रोटोजोआ आसीबो नै यूग्लिना जो थिला यूग्लिनॉइड्स आछथिला गोटे थिला किंगडम मोनेरा किंगडम प्रोटिस्टा प्रोटिस्टा कोन पांच प्रकार थिला क्रिसोफाइट्स डिनोफ्लैजिलेट्स यूग्लिनॉइड स्लाइम मोल्ड्स एंड प्रोटोजोआ तो जो यूग्लिनॉइड्स रे थिले यूग्लिना तो आर ना कोन हे गला दिस इज यूग्लिना और सेकंड ना कोन हे गला व्हाट इज द नेम ऑफ दिस ऑर्गेनिज्म आर ना कोन आर ना होची पारामीशियम दिस इज पारामीशियम और पारामीशियम कहा को बिलोंग करथिला ना हमर प्रोटोजोआंस को बिलोंग करथिला तो ये थिला यूग्लिना ये थिला पैरामीशियम तो यूग्लिना पाखरे कौन थिला यू कैन सी इन द केस ऑफ यूग्लिना दिस इज अ बेसल बॉडी एंड दिस इज द लॉन्ग स्लेंडर elongated flagella present over here flagellum जे तो गोटी अछि त ना हे गला flagellum long slender elongated flagellum is present in case of this euglinars and euglina mane thile they can do both type they can do or they are autotroph as well as heterotroph se mane nijo khadya nije banai parithile they are depending upon nobody nije nije depend करथिले otherwise they can be heterotroph और heterotroph कला माने se mane अबिका डायरेक्ट प्रेडेटर हे जाउथिले ସେମାନେ सीधा शिकार करी खाउथिले प्रेडेटर काम करथिले और जो autotroph थिले they are doing photosynthesis they are having their own cell chloroplast निज पाइथन chloroplast थिला they can do photosynthesis in the presence of sunlight and in the absence of sunlight they become predator से सीधा शिकारी हे कि मारी खाउथिले तो सेही मानन को आगु जीवा पई कि हेले they require a long slender elongated flagellum that is the structure नेक्स्ट असला पैरामीशियम कथा पैरामीशियम जो होची यू कैन सी पैरामीशियम ए पार्ट से जूम करके देखिए जी आप एखन दे टाइनी टाइनी बहुत सारा थाउजेंड हेयर लाइक पार्टिकल्स माने प्रेजेंट अछंती और जो हेयर लाइक पार्टिकल माने आउटगो थे बाहर को भारसन दैट इज कॉल्ड एज सिलियम गोटा कोइबा सिलियम बहुत सारा हे गले प्लुरल हेला सिलिया बोलता कोइ परिबे ए जो सिलिया माने अछंती आंकर काम हैला कोन काम हैला सेल को मूवमेंट करिबारे हेल्प करिवे सेल जो पार्टिकल जो जगह प्रेजेंट अच्छी सेठी का पानी रे जो खाद्य प्रेजेंट अछि खाद्य को निजर टाणिया पई ताकर इतिहास मोटिव होइसी और यू कैन सी जे जो मूवमेंट करूछनी किछि मूवमेंट अबिका ए डायरेक्शन आछि एंड देन से मूवमेंट फोन चेंज जाय कि उल्टा डायरेक्शन रिवर्स डायरेक्शन आगु बढ़ुची दिस इज कॉल्ड एज व्हिच काइंड ऑफ मूवमेंट दिस मूवमेंट इज कॉल्ड एज ओट्स लाइक मूवमेंट ताको हम कहबा कि ओट्स लाइक मूवमेंट बोली कहि परिबा Got it? Any doubt till this? बुझी पाइलो? Everything clear? Normally, yes? Okay. Done. Next segment को जीपा ये केमते तयारी हिचे ना कबे कोन कोन अरेंजमेंट हे गेछि ना ये जो रहिला हमरो बेसाल बॉडी गोटे रहिला बेसाल बॉडी और रहिला कोर ता ना हैला एक्सोनम दीटा पार्ट रे डिवाइड हिजंदी रिमेंबर ये जो हम पढुचे आज रे क्लास 3 दीटा पार्ट रे डिवाइड हिजंदी दिस इज द सेंट्रल कोर एंड दैट कोर इज कॉल्ड एज एक्सोनिम एको एक्सोनिम बोली कहै जे और तळे सेमन जोटो ओरिजिन करछनी दैट इज कॉल्ड एज द बेसाल बॉडी तो ये है तुम्हारा बेसाल बॉडी and this part is called as your axonym और these are structure different अच्छे तो our structure कौन है गला that is called as your very famous 9+2 arrangement what is 9+2 array और what is 9+2 arrangement ए word टे भल से बुझो पिला माने एठु तुम निहाती क्वेश्चन पाइबो तुम्हारा काउंसिल एग्जाम हो बा तुम्हारा एंट्रेंस एग्जाम रे हो देन देख पाछो तुमे ये सब हबा बाहर पटे के रहिला तार सेल मेम्ब्रेन टे प्रेजेंट अछि के प्रेजेंट अछि cell membrane or you can say it as the plasma membrane so बाहर पटी के रहला plasma membrane then ये plasma membrane ना दीटा जिंस रहला गोटे सेंटर देखो रहिची गोटे डबलेट टी जोड़ा जिंस मिसी गोटे डबलेट बनै चली ये गोटे सिंगल पार्ट दीटा मिसी के आराम से बनै जन ताको सेंटर रे अछि ना जगह सेंट्रल सीट आ चारि पर सेंट्रल सीट अछि कवर करी के अछि आ चारि पर जो देखियो त मतलब पेरिफेरी माना पाखरे दी प्रकार प्रोटीन स्ट्रक्चर अछि 1 and 2 suppose this is your B this is A this is B this is A this is B this is A and A भले माने एमपी 9 टा अछंदी तो हम काउंटिंग करे स्टार्ट कर this is 1 this is 2, this is 3, this is 4, this is 5, this is 6, this is 7, this is 8, and this is 9. So we're having 9 doublets around the periphery and center data singlet present achanti. So data singlet, that's why it's called as 2, and 9 doublet, that's why it's called as 9. So 9 plus 2 arrangement achanti. So A जो माने रहिले, this is called as tubule A, and tubule B एमन जो रहिले, they are called as a microtubule. ପିଲାମାନେ ମନେ ଅଛି ସାଇଟୋସ୍କେଲେଟନ जो ମାଇକ୍ରୋଟ୍ୟୁବୁଲ ପଢିଥିଲେ, ସେ ମାଇକ୍ରୋଟ୍ୟୁବୁଲ ଆମେ କଣ ଦେଖିଲେ 25 ନାନୋମିଟର ଲେନ୍ଥ ର ये माइक्रोट्यूबुल माने चिले दे आर मेड अप ऑफ अल्फा एंड बीटा ट्यूबुलिन प्रोटीन्स को नहीं समान तैयारी चिले और ये जो अल्फा और बीटा ट्यूबुलिन प्रोटीन्स माने अछंदी से ट्यूबुलिन प्रोटीन माने को अरेंजमेंट केमती हितला प्रोटोफिलामेंट टाइप रा अरेंजमेंट हितला ते मन रहे बीटा डबलेट आपस में केमिरी जॉइन हिचंदी बाय द हेल्प ऑफ नेक्सिन प्रोटीन प्रोटीन ना नाला नेक्सिन प्रोटीन जितना को हेल्प कर जॉइन करिबारे देन ता सहित आउ कोन अछि आंग पाखे नांग पाखे दी प्रकार रहिला डाइनिन प्रोटीन गोटे भीतर पटकू दीटा थिबो बाहर पटकू तीनटा थिबो डाइनिन प्रोटीन भीतर पटकू दीटा रहिबो बाहर पटकू तीनटा रहिबो डाइनिन प्रोटीन ठीक है दिस इज द आउटर डाइनिन प्रोटीन दिस इज द इनर डाइनिन प्रोटीन इनर पार्ट ए ब्लू कलर पार्ट को शो करछि दी प्रकार थिबो दीटा एंड बाहर पटकू तीनटा डाइनिन प्रोटीन माने थिबे देन आउट जिंस इंपोर्टेंट हे गेला कि ए जो पार्ट मझिरो भाजी दैट इज कॉल्ड एज रेडियल स्पूक और ना कोन हेगला दिस पार्ट इज कॉल्ड एज योर रेडियल स्पूक्स ये गोटे कार्ड टूल लाइक स्ट्रक्चर शो करछंदी तो ये सेंट्रल आर ना हेगला सेंट्रल सिच तो सेंट्रल सिच रहला मझिरे दिस आर द रेडियल स्पूक्स कनेक्टिंग the doublets into the central sheath is so doublet अछि मन क्यों doublet केटा अछि 9 टा अछि तो 9 into 2 is equal to 18 अछि but spoke केटा अछि spoke not ही अछि spoke 1 spoke 2 spoke 3 spoke 4 spoke 5 6 7 8 and of course your 9 तो 9 प्रकार रेडियल स्पोक प्रेजेंट अछंदी ए डायग्राम टा फरसे मन रखियो एनी डाउट इन दिस पार्ट बनमाली वर्षा मानस ऋतु प्रधान प्रज्ञा स्वीटी मिनर्भा मलिक any doubt till this anything you want to ask from this slide after completing this segment we'll quickly move to the any doubt you will ask this from this segment exam अछि okay all the best for exam चलो next segment asla cilia flagellary party cilium and flagellum are covered with plasma membrane the core is called as axoneme has many microtubules running parallel to the long axis and the axoneme has 9 pairs of doublets of radially arranged peripheral microtubules and a pair of central microtubule and this is called as 9 plus 2 arrangement. So dear participants, हम A part separation पढ़ुଥିଲେ. I need your involvement in this class. जो cilium और जो flagellum थिला, they are covered. तो तारी पढ़े क्या cover करेगी अच्छी plasma membrane, or you can say the cell membrane, like this. The cell membrane or the plasma membrane, this green color part is covering the entire sheath. I will say cover करेगी अच्छी. Number 2, their core. Core called the center जो part that is called as axoneme. It has many microtubules running parallel to the long axis. ठीक है जी ये parallel long axis पे मान सो मूव करते हैं यहाँ पे बहुत सारा microtubule मैंने अच्छे निकोड़ का axoneme central है. You can see the central microtubule केटा अच्छी डी टा अच्छी ये number 1 ये number 2 ଆଉ ଏମାନେ ପୁଣି ଆପସ ଜଏନ୍ ହେଇକି ଅଛନ୍ତି inter doublet bridge ଆଗ ଜାଗାରେ ସେମାନେ ଅଧିକା ବାଇଣ୍ଡ ହେଇକି ଅଛନ୍ତି। Then axoneme has 9 pairs of doublets. ये जो axoneme अच्छी central microtubule पर जो axoneme अच्छी केटा doublet अच्छी 9टा अच्छी कौन सी doublet doublet 1 doublet 2 doublet 3 doublet 4 doublet 5 doublet 6 doublet 7 doublet 8 and doublet 9. So 9 pairs of doublets are present at the outer side. These three are 1, 2, 3 outer side रहिले outer dynein. These two are inner dynein. मन रखिए part टा. ये जो 1, 2, 3 outer dynein, ये 4, 5 inner dynein. 1, 2, 3 outer dynein, 4, 5 inner dynein. तो एक तीनटार नाम जे देखिदबा that is called as outer dynein. ଆଉ ଏଇ ଯୋଉ ପାର୍ଟିକଲ ରହିଲା ପିଲାମାନେ ଆଙ୍କର ନାଁ କଣ ହେଇଗଲା these are called as inner dynein. Then एमाने आपस में join ହେଇଛନ୍ତି କାହାର help ରେ they are joined through the help of nexin protein. ये जो part ରହିଲା that is called as nexin protein. हमको क्या रिलेशन और आउट ना से कहला इंटर डबलेट ब्रिज बोली भी कुहाजे the nexin protein जो अछि it is also called as inter doublet bridge. तो ये रहा तुम्हारा central sheath. Centrally present it is called as central sheath. Sheath means covering and these are your radial spokes. So how many radial spokes you are getting here? There are 9 number of radial spokes are there. If we count from here, it's 1, it's 2, it's 3, it's 4, it's 5, it's 6, it's 7, it's 8, and it's 9. Here, we have 2 central microtubules. Then we are having a central sheath covering the microtubules. Then we have 9 outer peripheral doublets. And then these doublets are having interconnected bridge that is called as interdoublet bridge made up of nexin protein. Then they are having the outer dynein membrane. Dynein protein is the bahar photo tinta, visual photo data, outer dynein, inner dynein. And then they have a card-like structure formation. The peripheral doublets, the radially arranged microtubules, and the central microtubule. And this kind of arrangement is called as 9+2 array. This 9 doublet plus 2 centrally present microtubule. That's why it is called as 9+2 arrangement. So any doubt till this? Do participants want to ask any question from this? Most welcome, Mr. Ritesh Mohanty, Ritu Pradhan, Pragyan, Sweetie Digal, Manas, Varsha. Any doubt you want to ask, dear participants? This 9+2 arrangement of doubt in procedure chahiye ki? If you have any doubt, you can directly ask me in the chat box. Yes, any doubt till this? Okay. So again, say part asla phone amara. The central tubules are connected through the bridges and are enclosed by a central sheath. The ଚାରିପଟ ଘେରିକି ରହିଛି central sheath ଆଉ ଚାରିପଟ ଘେରିକି ରହିଛି ଆଉ ସେମାନେ connect ହେଇଛନ୍ତି central bridge ଦ୍ୱାରା. It is connected to one of the tubules of each peripheral doublet by a radial spoke, and thus there are 9 radial spokes present in the what? In the cilia or in the flagella. जो cilia और flagella जो हमें axoneme by the core part हमें देखुचे, से core part पर 9 टा radial spokes हैं. The peripheral doublets are also interconnected by the linkers that is called as linker protein. ये जो peripheral doublet माने अच्छे, this is a peripheral doublet, this is a peripheral doublet. These are interconnected by the help of linker proteins. ଆଉ ସେ linker protein ନା ହେଇଗଲା କଣ nexin. The name of the protein is nexin protein. Linkers बोलते କୁହାଯିବ. Cilia and flagellum emerge from centriole-like structure called as the vessel bodies. The cilia how, the flagella how, they all have been emerged from a central particle that is called as the vessel body. The vessel body is the so bahar ko bahar. Next, as like some different regular definition of cilia and flagellum, you can see over here. Cilia जो होची cilia are short hair-like appendages extending from the surface of a living cell. You got a living cell the surface ru extend ହେଇକି ବାହାରକୁ ବାହାରିଥାଏ, while the flagella they are long. Cilia is short, flagella are long. Cilium is singular, cilia is plural. Then flagellum is singular and flagella is plural. Yes or no? So the cilia are short hair-like appendages. They are extending from the surface of a living cell. But a living cell, the surface of an extend while the flagella are long thread-like appendages on the surface of a living cell. That means a long thread-like structure presentation. If you see the cross-section of a cilia or flagella, you will get Nexine arm is present in case of cilia, which is absent in case of flagellum. Yes or no? The cilia pakere nexine arm present achi, which is absent in case of our flagella. The length देखिबो तो cilia is of course short. हम आगुर पढ़ते cilia short थांती, and जो हम देखा flagella they are longer and they can vary from cell to cell, from organelle to organelle. But it is they are longer than the cilium. Now, which kind of motion they both are showing? In case of cilia, the motion is rotational motion, like a motor, like the very fast movement or like movement. But in case of flagellum, the motion is wave-like, undulating motion, the sinusoidal motion, the slow movement compared to cilia. Because the flagella को use करी कौन करची ना, से flagella को use करी के गोटे place रू आउटे place को जाऊची. लोकोमोशन କରୁଛି ତାକୁ ୟୁଜ କରି ସେ ଶିକାର କରୁଛି ପ୍ରେଡେଟର ହେଇଯାଉଛି ତା ପ୍ରେ କୁ କ୍ୟାଚ୍ କରୁଛି ତେଣୁ ସେ ତାର ଅବିକା ବାଲାନ୍ସ ମେଣ୍ଟେନ କରୁଛି କେତେବେଳେ ସେ ଅଣ୍ଡୁଲେଟିଂ ମୁଭମେଣ୍ଟ କରିବ କେବେ ସେ ତରଙ୍ଗ ହେଇକି ୱେଭ ଲାଇକ ମୁଭମେଣ୍ଟ କରିବ କେବେ ସେ ସ୍ଲୋ ମୁଭମେଣ୍ଟ କରିବ ଆଜ କମ୍ପେୟାର୍ଡ ଟୁ ଦ ସିଲିଆ ସିଲିଆ ମାନେ ବହୁତ ଫାଷ୍ଟ ମୁଭମେଣ୍ଟ କରୁଛନ୍ତି ମନେ ରଖିବ ତମେ ଡେନ୍ସିଟି ଦେଖିବ the number of cilia are many like hundreds per cell hundreds and thousands with thousands and thousands of cilia are present over a cell around the cell. But if you look at the bacteria, if you look for the flagella, they are having very less number, that is from 1 to less than 10 per cell. On the basis of presence or absence of flagellum, we have divided the bacteria into 4 different categories. One is having monotrichous, second is called as amphitrichous, third is called as lophotrichous, and the fourth one is called as peritrichous. Monotrichous mane kon hegala na single bacterium having a single flagella is called as monotrichous. Then single bacteria having 2 different flagella present on the opposite direction, that is called as amphitrichous. Then one single bacteria having a tufts of group of flagella present on one side, that is called as lophotrichous. And when the flagella are present all over the cell, all around the cell membrane, it is called as peritrichous. So if you talk about cilia, they are found in only eukaryotic cell. You can never find the cilia in prokaryotic cell. But in case of flagella, they are present in both prokaryotic as well as eukaryotic cell. Yes or no? I hope you may say if you have any doubt, then you can ask me in the chat box. We just right now completed the cilia and flagella portion. So we have completed the cytoskeleton, then the cilia and flagella portion is successfully completed. So the definition, the cross section, the length, the motion, the density, and the code each and everything I have discussed over here. Next, the cell organelles as well as centrosomes and centriole. This is the last part of this today's first class. And second class, I will start about the nucleus and all. Then the centrosome ki thala? The centrosome is an organelle containing 2 non-membrane bound cylindrical structure called as the centrioles, and they are surrounded by pericentriolar materials, and the centrioles lie perpendicular to each other They are made up of 9 evenly spaced peripheral fibrils or tubulin, and each of the peripheral fibril is a triplet, and the adjacent triplets are also linked. So, ये सब basic जिनसे हमारे देती regarding your cilia and flagella. तो तुमने देखी पड़ती हो जानी पड़ती cilia flagella कौन अच्छी ना cilia flagella सॉइला पर जो centrosome कथा आसिला पिला मैंने centrosome जो होची the centrosome होची गोटे organelle containing two non-membrane bound cylindrical structure called as centriole. तो दीटा सेंट्रियोल को नेकी बनछि गोटे सेंट्रोजोम तुम देखि पारछो पिक्चर राइट साइड रे दीटा सेंट्रियोल प्रेजेंट अछंती एंड दे आर परपेंडिकुलर टू ईच अदर पिला माने एई पॉइंट निहाते मन रखिबो. Please remember this point, like the centriole do detachment, the centriole are perpendicular to each other. The centriole lie perpendicular to each other and they are made up of 9 evenly spaced peripheral fibrils of tubulin. तो ସେମାନେ କାହାକୁ ନେଇ ତିଆରି ହେଇଛନ୍ତି ନଅଟା evenly spaced peripheral tubulin protein ତିଆରି ହେଇଛନ୍ତି. ଏଠି 9+0 arrangement ଅଛି and each of the peripheral fibril is a triplet in nature and the adjacent triplets are also linked. और जो ताऊ side जो adjacent जो side by side जो triplet बना अच्छे से माने कि आपस में join है कि अच्छे link है कि अच्छे. Any doubt till this? You wanna ask? The participant, any doubt you wanna ask from here? You can see over there, these are the 3 triplets. 1, 2, 3. ये तीनटा मिसी वो ट्रिपलेट बनेले. पुणी एटी 1, 2, 3 तीनटा मिसी तीनटा ट्रिपलेट बनेले. Then 1, 2, 3. And A भड़िया triplets बने मिसी की 9 टा triplets presentation. 1, 2, 3. Again, this is 1, 2, 3. So 9 triplets are there. If you count, this is triplet 1, triplet 2, triplet 3, triplet 4, triplet 5, triplet 6, triplet 7, triplet 8, and triplet 9. The 9 triplets are present in a single centriole, and if this is a single centriole, then it is a single centriole, and that is called as centrosome. This particular part is called as centrosome, and the centrosome जो रहिला ये centrosome केमती arrange ହେଇକି ना एमाने present अछंती 9+0 arrangement T shape रो देखिया ପାଇଁ कि perpendicular to each other. They are perpendicular to each other. That means having a 90-degree angle in between them. Clear? Any doubt in this? Then the central part of the centriole is also proteinaceous and called as the hub, and which is connected with the tubules of the peripheral triplets by radial spokes. And what radial spoke mane present achindi. Also radial spokes jo rahila the centriole from the basal body of the cilia or flagella. They are spindle fibers that give rise to spindle apparatus during the cell division in case of animal cells. The central part जो रहला centriole that is called as proteinaceous hub बोल तक हमें कहि पारिबा central part को. आउ से central part पाखरे कोन अछन tubules अछंदी आउ radial spokes अछंदी. जो radial spokes माने कहा से कनेक्ट हेक अछंदी तार triplets से कनेक्टेड अछंदी which is made up of a kind of protein. Then the Centrioles form the basal body of the ciliar flagella and the spindle fiber that give rise to the spindle apparatus during the cell division in case of our animal cell. The animal cell division bole emane help kartani. Now it's time to attend some question and answer. The dear participant, if you are not going to participate in the question and answer segment, then I cannot found or justify that you have understood the entire class. So, Sweetie, Pragya, Nitu, Ritesh, Manas, Varsha, I'm requesting all the participants to kindly participate and answer these very few basic questions. The cytoskeleton is primarily composed of calcium carbonate granules, proteinaceous filaments, cellulosic microfibrils, and callose deposits. What are you going to answer this question? Yes, dear participant. Yes, answer to this first question. The cytoskeleton is made up of which filaments? Is it calcium carbonate granule? Is it proteinaceous filament? It is which is cellulosic microfibrils, or it is callose deposits? What is the right answer? Yes, what is the right answer? The right answer is proteinaceous filament. Yes, the participant, what are the right answer? The cytoskeleton is made up of basically the proteinaceous filament. Answer ala proteinaceous filament. The proteinaceous filament रे क्या आस्थिला तीन प्रकार part आस्थिला. If you remember, one is your microtubules, second is intermediate filaments, and third is the microfilaments. And your tubulin protein, your actin protein, your keratin protein. This is your tubulin protein. This is your keratin or vimentin or lamine protein theory hitandi, and this is made up of your actin proteins. ठीक है तो आत्मी नहीं हमें तैयार करते देखते पार्ट की क्वेश्चन नंबर 2 which of the following is not a part of cytoskeleton बोल से देखिता which of the following is not a part कोटा है चलो cytoskeleton ना part नहीं yes finally I got an answer from an for a participant. Thank you so much for joining. Which of the following is not a part of the cytoskeleton? Cytoskeleton part किए नहीं. This is a part microtubule. This is a part microfilament. This is a part endofilament. Which one is not a part? Yes, option D is correct, Nitu. The right answer is microfibril. These are not a part of this microfilament. Question number 3. Microtubules are constituents of dash. These microtubules कहाँ को नेकी तेरी चने कहाँ रा part रे हम देख परिबा. Microtubule को जगह माने present अच्छी. Spindle fibers, centriole and cilia. Centriole, spindle fibers and chromatin. Centrosome, nucleosome and centrioles. Cilia, flagella and peroxisome. तो चार्टा ऑप्शन रो हमें पेरोक्सिज्म पे किसी तो पढ़ी नहीं टिल ना आ कथा कोठी भी उठिनी ताको कैंसिल करो। देन सेंट्रोजोम न्यूक्लियोजोम सेंट्रियोल सेंट्रियोल हमें पढ़िले सेंट्रोजोम पढ़िले न्यूक्लियोजोम तो पढ़िने तो आको भी कैंसिल करो। देन सेंट्रियोल स्पिंडल फाइबर्स सेंट्रियोल पढ़िले स्पिंडल फाइबर पढ़िले हेले हमें क्रोमैटिन कथा कोठी भी पढ़िने टिंडा तो आको भी कैंसिल करो। The right answer is your option A, which contain the spindle fiber, the centrioles and the cilia. The cilia रे भी अच्छी, centriole रे भी अच्छी, spindle fiber रे भी अच्छी. किए अच्छी? Microtubule. The microtubules माने कहाँ constituent अच्छी? They are the constituents of the spindle fiber, the centriole and the cilia. Next, the motile bacteria are able to move. तो बैक्टीरिया माने जो मूवमेंट कर पारछने मोटाइल माने मूवेबल बैक्टीरिया. The motile bacteria are able to move. Yes. Yes, I got an answer. Replacing option C. Yes, the correct answer is option C. This correct answer is option. Sorry, option C is not the correct answer. The motile bacteria able to move. Move move mane gote jagah flagella is the correct answer. The correct answer is flagella. The correct answer is flagella is the option B is the correct answer. The characteristics pattern arrangement of microtubule in the axoneme of a cilium is axoneme arrangement केमतिया हेइतिला. Option D, that is 9 plus 2 arrangement. Yes, 9 plus 2 is the correct answer. Then 5 more questions. The core of cilia and flagella which contain the microtubule that is called as core. That core part is basically known as the axoneme. Core part is basically called as axoneme. Both cilium and flagellum arise from a part structural similarity to centriole, kinetochore, kinetosome, centrosphere. Now answer the question. Yes, I got an answer in the chat box. Ritu is saying option A is correct. Okay, option A. The both cilium and phloem arise from a part structurally similar to, yes, option A is correct. That is the centriole. Absolutely correct. Yes. Next question. The centriole-like structure. The centriole-like structure from which the cilium and flagellum emerge. जोटो cilium और flagellum टी emerge करची बाहर को बाहरची is called as centrosome, basal body, axoneme और hymentin. ଯେମିତି ये ହେଇଗଲା core part. तो ये कोड origin कोड करची. From where does it get originated? It get originated from option B, that is the basal body. Next question. Which of the following is a correct statement regarding the axoneme of eukaryotic flagella? Eukaryotic flagella axoneme go to TDG. Which of the following is a correct statement regarding the axoneme of eukaryotic flagella? It is composed of 9 peripheral triplet and microfibrils. Axonium flagella axonium कथा कही vessel body नुहे. Peripheral triplet wrong. It does not have a covering of plasma membrane. It do have. So it be wrong. It gives rise to the spindle apparatus. Option D. Again wrong. The right answer is central tubules are enclosed by a central sheath. The center रे जो ट्यूबुल माने थिले ताको गोटे सेंट्रल शिफ्ट कवर करथिला मन पडुची आ चारिपट सेंट्रल शिफ्ट थिला एठी समस्त डबलेट माने प्रेजेंट होतिले डबलेट माने होतिले लास्ट क्वेश्चन cilia and flagella resemble each other in cilia and flagella each other केमते resemble करछन्दी microtubular structure correct presence of basal granules correct mode of action eight are incorrect so option D जो होचे more than one option is correct yes more than one option is correct अछि कहीं A and B both are correct these are तो मन अपन के homework is the part so thank you so much for your kind attention and we'll meet again after a 15-minute break the class will resume from 15 minutes 9:15 AM रे हम क्लास को resume करियो Welcome once again, dear participants. We will resume the class from here. We are going to the second class, and the second class is all about, we are going to study about nucleus, nucleolus, chromosomes, and then the microbodies. ठीक है हमारे चार्टा पार्ट जिन पढ़ार अच्छी nucleus, nucleolus, and the chromosomes and microbodies in this class. Once again, I am Devashish Sahu, lecturer in botany, and I welcome you all to this DHSA sponsored online academic support program class, where we are going to give a detailed analysis about the chapter cell, and this is the last part of this chapter, and we are going to expect 1 to 2 questions around from this segment, because it's the brain of the cell. We are going to discuss about the nucleus. Now, so first thing first, what do you mean by nucleus? Nucleus was first described by Robert Brown. ठीक है जी, तो nucleus को क्या discover करते हैं? The name of the scientist who discovered nucleus is known as Robert Brown. You all know him, and he discovered the nucleus in 1831. तो nucleus discover ହେଇଥିଲା 1831 ରେ discover କରିଥିଲେ scientist का नाम होची Mr. Robert Brown. Okay. The material of the nucleus stained by the basic dyes were given by the name chromatin, and the chromatin now the scientist का नाम है Fleming. ठीक है जी, the scientist Fleming who was given the name chromatin और उसे basic dye रे नाम आस्तेला. Whenever I say chromatin, ये जो chromatin word अच्छी chromatin word the word chrome, that word chrome ता माने हेगला कौन है ठीक chrome माने हेगला colors, chrome माने कौन हेगला colors. The chromatin by Fleming. Normally, cell has only one nucleus. Some cells have more than one nucleus. Maybe gote cell pari gote nucleus hoy paribo, or maybe some cells have more than one nucleus. That condition is called as xenocytic. One cell usually has one nucleus. Nucleus is the brain of the cell. But if a single cell is having many nucleus, बहुत सारे nucleus रहिला, many nucleus is there, then that condition is called as xenocytic condition. That condition is called as xenocytic condition, or you can say it is as multinucleate condition. You can call it as multinucleate condition. Next, some mature cells lack nucleus. किसी cell में nucleus नथा है, जेमती कि example is the mammalian RBC in case of animal cell, and in the sieve tube cells of the vascular plants. But whenever I say vascular plant, the vascular plant रे को माने कथा आसिला vascular bundle ले आसबो कहा कथा. We are going to talk about the xylem and phloem. तो जो xylem है गला by जो phloem है गला आंको पाखे जो sieve elements माने अच्छी. The sieve elements, sieve tube, these are dead cell basically. और आंको पाखे कौन नहीं, nucleus is absent. Nucleus is absent. Not that. Clear? ଆଉ ଯଦି ମୁଁ କହିବି ଏଠି ପ୍ଲାଣ୍ଟ ନହେଇକି animal cell ଦେଖିବି, then animal ମାନଙ୍କ ପାଖରେ କଣ ଅଛି, blood ଅଛି. Animal cell ରେ present ଅଛି blood. ଆଉ ଏ ଯୋଉ blood ଅଛି, it contains RBC. ଆଉ ଏ ଯୋଉ RBC ଅଛି, the mature RBC ପାଖରେ nucleus କଣ ଥାଏ, nucleus ନଥାଏ. Nucleus is absent in case of RBC. That is the thing. If you see a nucleus, you can clearly see that this nucleus is a double membrane structure. Am cell emti tinta jinsa achani which are made up of double layer, and the answer is 3. NMC. NMC. What is N? N is nucleus. What is M? M is mitochondria. And what is C? C is your chloroplast. So in the NMC, nucleus, mitochondria, and chloroplast, all these three are double membranous. All these three are basically double membranous. Across data membrane को नहीं हमारे theory ही था थी. ये क्या nucleus, mitochondria और chloroplast. जो जिन double membrane अच्छी, of course the outer one is called as outer membrane, the inner one is called as inner membrane, and in between the two comes a space that is called as perinucleolar space. That is called as perinuclear space. I hope तुम्हें इतनी जिन सब बुझी पाइला. Scientist Robert Brown in the year 1831 has discovered the nucleus central of the brain of the cell nucleus, which is called as your brain of the cell. ये जो न्यूक्लियस अच्छी से न्यूक्लियस पाखरे कौन अच्छी ना specific dye the stain कराइला that is called as chromatin thread like structure chromatins are there. And normally a cell has one nucleus, the type unicellular, uninuclear, or the multiple nucleus that is called as multinucleate condition, or you can call it as xenocytic in nature. And किसी पाखरे तो बिल्कुल nucleus ही नहीं that is called as lack of nucleus in case of mammalian RBC, unsieved two cells of vascular plants. I hope you got it. So if any doubt, you can ask me in the chat box. Next, the interphase nucleus contains the four parts. One is the nuclear envelope, second is the nuclear matrix or the nucleoplasm, third is the chromatin, and fourth is the nucleolus. The interphase nucleus, ये जो हमारा nucleus जो interphase nucleus हम जो cell division time में गोटे phase ऐसे का नाम है ना interphase. तो आम सेल्स डिवीजन टाइम कला बेले माइटोसिस मेयोसिस ये सब जिनसे कंटीन्यूअसली चलता है. और माइटोसिस मेयोसिस रा जो फर्स्ट पार्ट है दैट इज कॉल्ड एज इंटरफेज, जोटे सेल जो प्रिपेयर करूची कि आगु जाय से डिवाइड करियो बोली. और से डिवीजन टाइम रे इंटरफेज टाइम रे सेल भीतर कोन फॉर्मेशन हेता है, सेल फॉर्मेशन हेता है तार डीएनए डुप्लीकेट करता है G1 फेज, S फेज, G2 फेज. और से सब बेसिकली हेगला इंटरफेस. In the interface the nucleus contains nuclear envelope, nuclear matrix that is called as nucleoplasm, then chromatins, of course the thread like structure, colorful thread like structure, and the last part that is called as nucleolus. तो कौन कौन जिनसे रहिला चारटा पार्ट रहिला. one is a nuclear envelope. एनवेलप जो जिनसटी कहले पिला माने से चारिपट दा जो बाउंड्री टे अछि जो ता कवरिंग टे अछि डबल मेम्ब्रेनस कवरिंग. देन द न्यूक्लियर मैट्रिक्स देन द सेल केस रे साइटोप्लाज्म, माइटोकॉन्ड्रिया मैट्रिक्स, क्लोरोप्लास्ट स्ट्रोमा, सेमती न्यूक्लियस रो न्यूक्लियोप्लाज्म बा न्यूक्लियर मैट्रिक्स ता कहिबा. देन ता भीतर अछि थ्रेड लाइक स्ट्रक्चर दैट इज कॉल्ड एज क्रोमैटिन्स. एंड इन द लास्ट सेगमेंट द न्यूक्लियर विस्तृत है कि थांदी न्यूक्लियोलस माने थांदी. डन. और ये जो न्यूक्लियस रो जो बाहर पटे एनवेलप अछि से एनवेलप को कंटिन्यू करी को माने अछंती एनवेलप को कंटिन्यू करी प्रेजेंट अछंती तार एंडोप्लाज्मिक रेटिकुलम माने. That endoplasmic reticulum is again of two types. One is a rough endoplasmic reticulum and the second one is smooth endoplasmic reticulum. तो ये endoplasmic reticulum माने present है ता भीतर. Yes, any doubt आ भीतर किछि you want to ask? Okay, right now I'm having 6 more participants. So, any doubt? The participants, the interface nucleus.
सबनु बेसी इंपोर्टेंट रहला एंट्रेंस. छोटा कर देनी सुनो हैं कुप्रों तो देखिए तुम ओ यूएटी बोलू ने.
Okay, so the participants kindly mute yourself so that we'll proceed. So an humble request to all the participants who have not muted, kindly mute yourself.
लिखो लिखो ओडिशा यूनिवर्सिटी ऑफ एग्रीकल्चर एंड टेक्नोलॉजी
Okay. So dear participants, now kindly focus on this following part. So in this nucleus segment, we are discussing about the interface nucleus and its contains the four different parts. That is the nuclear envelope, the nuclear matrix, the chromatin, and the nucleolus. You all know it very well from the school days. That is the nuclear envelope is nothing but the nuclear membrane. And this nuclear membrane is a double layered structure. It's a double layered structure which contains both outer membrane as well as inner membrane. Then it has both outer membrane and other as also it has inner membrane. Clear? Then the nuclear matrix, then the cell ଭିତରେ cytoplasm, so the nucleus ଭିତରେ কি ଅଛି nucleoplasm present ଅଛି. Then the chromatin, the colorful thread-like structures. The colorful thread-like structures that is called as a chromatin, and then of course last the nucleolus, you have the ribosome factory. Nucleolus are called as the ribosome factory. Why it is called as ribosome factory? Who is going to answer this question? Pilamane, dear participants, who is going to answer this question? Now we are having 8 more participants. So my question is to all of you, like, why these nucleolus are considered as the ribosome factory? We all know ribosome is the protein factory of the cell. Ribosome nije hochi protein factory. It helps to make proteins. Yes or no? The ribosome is protein factory, but the ribosome who kiye banaychi? How this ribosome is made up of? You all have written. To answer this question, we all know that ribosome is the protein factory of the cell. And where does that ribosome is made up of? Ribosome both information you see. The ribosome is made up of two things. One is your ribosomal RNA and second is ribosomal proteins. So your ribosomal RNA, or which is called as simply rRNA, that rRNA is made by whom? That rRNA is made by this nucleolus. So inside the cell there is a nucleus. Inside the nucleus there are some colorful thread-like structure chromatin. So मैंने chromosome formation करचंदी. और से जो chromosome को जो chromatin अच्छी से chromatin आगो जाएंगी कौन करची ना ताकि से formation कर दो ची RNA. और RNA जो पिला मने थिला हमारा RNA 3 type ର थिला. Eukaryotic cell 3 type RNA थिला. गोटे हैला rRNA, गोटे हैगला mRNA and गोटे हैगला tRNA. So ये basically 3 different types of RNA थिला. rRNA is called as your ribosomal RNA. It's called as ribosomal RNA. mRNA is called as your messenger RNA. And tRNA called as your transfer RNA. Then this 3 RNA actually. Our body is 100% RNA देखिबा. Out of this 100% RNA, our distribution are देखिया पिला माने 3टा part के divide ହେଇचंदी. और 3टा part किए ना 80% of the total cellular RNA is occupied by rRNA. Then 15% of the total cellular RNA, 15% part को क्या occupy करची tRNA. And rest 5% को क्या occupy करची your mRNA. So you can say out of 100% of RNA, 80% RNA is your rRNA, that's called a structural RNA. And the structural RNA को किए बनेची nucleolus is making. जाको तुम ओडिया रे नेस्टी बोली कहउथिलो nucleolus ସେଇ ଜିନିଷଟି ବନେ that's why it is called ଆଉ ଏଇ ଯୋଉ rRNA ଅଛି this rRNA is a part of ribosome ଆମେ ଯୋଉ ରାଇବୋଜୋମ ପିଲାମାନେ ଜାଣିଛେ ପ୍ରୋଟିନ ଫ୍ୟାକ୍ଟରି is called as basically ରାଇବୋଜୋମ ସେ ରାଇବୋଜୋମ ପାଖରେ ପିଲାମାନେ ଦି ପ୍ରକାର ଜିନିଷ ଅଛନ୍ତି ଗୋଟେ ହେଲା ତାର ରାଇବୋଜୋମାଲ RNA ଆଉ ଗୋଟେ ହେଇଗଲା ରାଇବୋଜୋମାଲ ପ୍ରୋଟିନ राइबोसोमल आरएनए जो रहला rRNA आको ही नहीं जैसे तैयारी ହେଇଛି that's why it is called as the ribosome factory of the cell is it clear ତମେ ତ ବୁଝ ପାଇଲ କଣ କହିବାକୁ ଚାହୁଁଥିଲି ଏପର୍ଯ୍ୟନ୍ତ any doubt you have any doubt you can ask me in the chat box the participants Arjun Higher Secondary School and MJ Higher Secondary School Sambalpur the participants if you have any doubt you can unmute anytime and you can ask your question directly or you can take the help of chat box and you can post your question there also then Sahil Pragya Pankaj Ritesh anytime you got any doubt you can raise your hand you can ask me questions in the chat box or you can ask questions directly by unmuting yourself then is it clear till this भलसे बुझ पाइलो आम बॉडी रे जो सेल अछि सेल रे जो न्यूक्लियस अछि न्यूक्लियस में चारटा पार्ट देखेली इंटरफेस ऑफ न्यूक्लियस चारटा पार्ट अछि व्हाट आर द फोर पार्ट्स नंबर 1 is your nuclear envelope that is the outer membrane and the inner membrane number 2 is the matrix of the nucleus which is called as a nucleoplasm number 3 it is the chromatin the thread like structures and number 4 is of course here the nucleolus which is called as the ribosome factory of our cell because ribosome जब रहिबनी ताहेले हम protein formation हे परिबनी and to make protein we need ribosome to make ribosome we need rRNA and to make rRNA we are dependent upon the nucleolus which is present inside the nucleus that is all I want to convey you to this online academic support program now you can see very well a very bright, very beautiful structure of the nucleus. All you can see here, what is the nucleus and how it is made up of. You can all see this nucleus is having two membrane. One is the outer membrane which is pink in color. Second is the inner membrane which is purple in color. ठीक अछि तमे दीटा कलर जे पिला माने देखि पड़छ गोटा आउटर मेम्ब्रेन टे अछि पिंक कलर रा गोटा वायलेट वा पर्पल कलर रा अछि inner membrane you can all see. In between the two nucleus, in between the two membrane, there is a space which is known as perinuclear space. And that perinuclear space जो present अछि ता काम हैला कोन ना न्यूक्लियस भितर जो भी जिंस जाउछि बासुछि ताकर ट्रांसपोर्टेशन को तार कम्युनिकेशन को कंट्रोल करिबा ट्रैफिकिंग करिबा रेगुलेट करिबा again out with a green color layer तुमने देखि परछ पिला माने that green color layer is nothing but it is a nuclear lamina तो न्यूक्लियस रो जो मेम्ब्रेन सहित माने लागि थांते लैमिन प्रोटीन तयारी हितने that's why it is called as nuclear lamina बोले आपको कहितो then मझरे मझरे न्यूक्लियस रो जो मेम्ब्रेन मन हम देखिबो दे पैरेलल टू ईच अदर आ गुडे जागर समान आपस में फ्यूज हे जैचंदी एंड सेटी गोटे स्पेस टे रह जायछि that space is nothing but called as a nuclear pore the space is called as nuclear pore ଆଉ से nuclear pore देकी nucleus रे बनिथिबा जिंस माने बाहर को आसते now you will ask question nucleus रे सर कोन जिंस बनता है ना nucleus रे बनता है को माने ना RNA are formed inside the nucleus DNA is there and so DNA अछि it can go outside to the ribosome directly to produce protein so it rather sends a messenger and that messenger is a nothing but it's an RNA और से जो RNA अछि messenger RNA जब से बाहर को जाय छै से जीवा पाई गोटे बाटो रखि थाय से बाटो ना हैला चैनल ना हैला न्यूक्लियर पोल and you will see the brown color rounded circular structure which are present around the surface of the pink color part that is nothing but the ribosome and that ribosome are the protein factory of the cell. These ribosomes are not only present on the nuclear membrane, they are also present on the endoplasmic reticulum. And that's why this endoplasmic reticulum surface gets rough and hence it is called as rough endoplasmic reticulum or RER. This rough endoplasmic reticulum has a very basic property to formation of proteins. एमाने protein formation करी पारंती एते जिनस भलसे मने रखिबो. Is it clear? Any doubt till this you can ask me in the chat box. Your queries, your feedbacks are very important, participants. Any doubt till this you can ask me in the chat box. Okay, I got the chart. Okay, no doubt. Good. So we'll follow from here. We'll start from here. So welcome once again. So this is the outer membrane. This is the inner membrane. This is the nucleolus, and in between you see a dense protoplasmic dense matrix is present, which is called as your nucleoplasm. Inside the nucleoplasm, what is happening basically inside the nucleoplasm? The DNA gets replicated, and then the new DNA gets transcribed. And when transcription occurs, what we got RNAs from those DNA. So our DNA ଭିତରୁ DNA ବନିବ, process is called as replication. Then say DNA ରୁ RNA ବନିବ, the process is called as transcription. And both the process are occurring inside the nucleoplasm. This blue color centrally present part is called as the nucleolus. ଆଉ जो न्यूक्लियोलस अछि पिलामाने न्यूक्लियोलस ର କାମ ହେଲା କଣ ये तुमरा आरआरएनए फॉर्मेशन करथांती आरआरएनए जोटा कि आगो जाईकी तुमरा राइबोसोम को फॉर्मेशन करिवो आई होप ए जाई बुझी परथिबो स्टिल किछि डाउट रहले यू कैन आस्क मी इन द chat box now you see the nuclear envelope. It is a double layered membrane with a space between 10 to 50 nanometer called as the perinuclear space. This one, the perinuclear space of kete gap achi 10 to 50 nanometer. This perinuclear space is having a space of around 10 to 50 nanometer, which is called as peri. Peri means the periphery, outer side, nuclear space. It is a barrier between the materials present in the nucleus and in the cytoplasm. You can see this membrane jo rahila. Outside the membrane, they are the cytoplasm. Inside the membrane, they are the nucleoplasm. So in the cytoplasm or nucleoplasm, मझिरे गोटे barrier रहिला से barrier ना हेगला कण that is called as nuclear envelope. Also, nuclear envelope जो रहिछि that is having two part. One is the outer membrane, second is the inner membrane. And inside the two membrane, in between the two membrane, a tiny space is present that is called as perinuclear space, which is having 10 to 15 nanometer width. ହେଇପାରିବ କିଛି କିଛି ଜାଗାରେ ହେଇଛି କଣ the outer membrane usually continues with the endoplasmic reticulum and also bears ribosome in it. तो ଏଇ जो outer membrane ଅଛି ତାରି ପାଖରେ କୋଉ ମାନେ ପ୍ରେଜେଣ୍ଟ ଅଛନ୍ତି ନା endoplasmic reticulum ଅଛି. You can see this endoplasmic reticulum and you can see the choco chips like structure that are called as the ribosome. ये जो गोले गोले brown color pigment you can see that are nothing but the ribosomes. ये जो ribosomes माने अच्छी ribosome दी प्रकार structure अच्छी. One is small subunit and large subunit. Small is basically here is called as 40s. Large here is called as 60s, and in total they are called as 80s. ठीक है तो ये जो राइबोसोम अच्छे स्मॉल सब यूनिट एंड लार्ज सब यूनिट एमन आपस में मिसकी फॉर्मेशन करछन गोटे राइबोसोम को आ से राइबोसोम आगो जाइए प्रोटीन फॉर्मेशन करिवो प्रोटीन बनिया पाएगी ता मध्ये कि आसीबो एमआरएनए दरकार आ से एमआरएनए बाहर को बाहर जी को बाट देकी बट एमआरएनए इज coming outside of the nucleus through this nuclear pore. ये जो nuclear pore अच्छी ये nuclear pore देके हमारे बाहर को बाहर की आसवा. Next, the nuclear envelope has minute pores formed by the fusion of these two membranes. जो membrane आपस में fuse हुए, and these are the passages for the movement of RNA. RNA कहले को RNA बुझौछि, RNA कहले तीनटा RNA कथा पड़छि. गोटे गला mRNA, गोटे गला rRNA, आ गोटे गला tRNA. सब RNA मानकर मूवमेंट पे माने हेल्प करतंदी एंड प्रोटीन बिटवीन न्यूक्लियस एंड साइटोप्लाज्म. अगेन प्रोटीन फैक्ट्री किया हमर अछि राइबोसोम. राइबोसोम कोठी प्रेजेंट अछि आउटर सरफेस रे. जैसे आउटर सरफेस रे प्रोटीन फॉर्मेशन हबो, देन प्रोटीन फॉर्मेशन हे सारला परे द सेम प्रोटीन विल कम इनसाइड द न्यूक्लियस थ्रू द न्यूक्लियर पोर. तो ये जो न्यूक्लियस चारिपटे जो न्यूक्लियर पोर प्रेजेंट अछि ये न्यूक्लियर पोर दे कोन ट्रांसपोर्टेशन हे परछि. कोन ट्रांसपोर्टेशन होछि ना आरएनए जाउछि बाहर को, प्रोटीन आसुछि भीतर को. And when I say protein, which kind of protein I am talking about? I am basically talking about histone protein. And DNA को bind करिया पाएंगे, DNA को condense कर रखिया पाएंगे, we need the help of a kind of protein that is called as histone protein. Which protein? It is called as histone protein or histone octamer बोल तक हमें कहिता. So remember this segment पिला माने ए जाय बुझी पारलो. Till किसी doubt रहुछि, you can again ask me. Then we'll go forward to the chromatin wala segment. What is chromatin? Now we're having another participant, Janta Higher Secondary School in Dhenkanal. So now let's again begin from this chromatin wala segment. Chromatin जो part वाला पिला माने, it is a network of nucleoprotein. थोड़ा जिनसो नहीं तैयारी जी गोटे ला nucleic acid and second is the protein. न्यूक्लिक एसिड कहले कोन बुझछो, न्यूक्लिक एसिड कहले डीएनए कथा कहूची और प्लस ता सहित के रहिला प्रोटीन रहिला. दुईटा का आपस में मिसी गले कोन फॉर्मेशन करचंती क्रोमैटिन बनाउचंती. जेने न्यूक्लिक एसिड रे आसिला डीएनए सेने प्रोटीन ने को प्रोटीन आसीबो. Basically we are talking about histone protein or histone octamer. What is histone? Now histone is a type of protein which is basically five types of histone thanti. One is your H1 histone, then your H2A, then comes H3B, sorry H2B, then comes H3, and the last one is H4. Among those five histones, these four histones all together produce a histone octamer like this. One histone protein, second histone protein, third histone protein, fourth histone protein. So ये वाला 1, 2, 3, 4. Another 4 ता पछपटे. Again 1, again 2, again 3, and again 4. And it successfully makes a histone octamer. We have successfully make a histone octamer. ଆଉ ଏଇ ଯୋଉ histone octamer ଅଛି ଏ histone octamer ଚାରିପଟେ କିଏ ରାଉଣ୍ଡ କରିବ ନା DNA wrap କରିବ. DNA ଆଉ ଯୋଉ DNA wrap କରୁଛି DNA plus this histone octamer ଯଦି ମିଶିଯିବ then it is called as DNA plus your histone protein is called as your nucleosome. This is called as nucleosome. ଆଉ ଏଇ ଯୋଉ ନ୍ୟୁକ୍ଲିଓଜୋମ ମାନେ ଅଛନ୍ତି ସେ ପୁଣି କଣ୍ଡେନ୍ସ ହେବେ, ଆହୁରି କଣ୍ଡେନ୍ସ ହେଇକି ଆଗକୁ ଯାଇକି ଫର୍ମେସନ କରିବେ ଲାଇକ୍ ଦିସ୍ ଷ୍ଟ୍ରକ୍ଚର. And it called as chromatins. आपको यहाँ में chromatin बोले कौन-सा? Clear? So it contains DNA. So जो हम nuclear protein का nuclear protein ने क्या है? It contains DNA and it contains a basic protein called as your histone. My question is why it contains a basic protein? कौन पाएंगे basic protein रखेला? Why not acidic protein? Why basic protein? So participant, my question to all of you. Janta Higher Secondary School, MJ Higher Secondary School, Sambalpur, Ritesh, Pankaj, Arjun High Secondary School, Sahil, Pragya, question is to all of you. Why we have given basic protein? Why the histone proteins are basic proteins? Why not acidic proteins? Yes, quickly answer this question. If you answer this question, then I'll be very happy. If you can't answer this question, then you can say, sir, we have a doubt in this segment, then I'll let you clear your doubts. Any doubt there, participants? Why we are having basic histone protein? Why not acidic histone protein? Any doubt? Anyone having any doubt? Yes, we got an answer. Okay. Thank you so much for your kind concern. Then comes our nucleus part. So it is nothing but one or more non-membranous spherical bodies that is called as nucleolus. It is continuous with the nucleoplasm and the site for ribosomal RNA synthesis. Before going to the nucleoplasm part, मैंने जो DNA अच्छी, if you see DNA, DNA is deoxyribonucleic acid. Deoxyribonucleic acid. First of all, it is acidic in nature. It is acidic in nature. And DNA को wrap କରିବାକୁ ହେଲେ I need a basic protein. Why so? The DNA is acidic in nature as well as it has phosphate. As it has phosphate or phosphoric acid, that is PO43 minus. So the overall charge of nucleus. So the overall charge of nucleus, the overall charge of DNA is negative. जब हमको डीएनए को बाइंड करने पड़े सो व्हाई वी आर पैकेजिंग दैट डीएनए हम डीएनए को पैकेजिंग कौन बाइंड करछो व्हाई वी आर पैकिंग दैट डीएनए इफ यू सी इन केस ऑफ यूकैरियोटिक mammalian cell the length of DNA is the length of DNA is 3.4 into 10 to the power 9 multiplied with 0. Sorry. Directly you can write DNA is 2.2 nano 2.2 meter. Finally, approximately 2.2 meter. The 2.2 large structure DNA को हमको रखिया को हेले bind को रखिया को हेले we need a protein. That protein should be acid जेहेतु अच्छी तो acid रो opposite से basic हेतु दरकार and जेहेतु ये negative अच्छी तो negative against से कौन सा positive हेतु दरकार. So we need a protein which should made up of it should be made basic and positively charged basics and positively charged amino acid आपको इन्हें के तैयारी देते हो basics and positively charged amino acid से हेतिबो और सेम ते हम पाखे जिंस अछि कि that is called as lysine and arginine से amino acid असले किए now one is lysine second हैला arginine और lysine plus arginine आपस में मिलके बनै जने कोन हिस्टोन ऑक्टामर बनै छै clear तो basic overview तम पाई गला DNA / deoxyribonucleic acid which is acidic in nature as it has phosphoric acid as a sugar phosphate backbone so the overall charge of DNA is negative so DNA को bind करिया पाई का हमरे एमते प्रोटीन दे दरकार which should be basic in nature and is having positively charged so जो positively charged basic amino acid हम पे जीटा अछि one is lysine and second one is arginine और lysine and arginine आपस में मिलके फॉर्मेशन कर देबे कहार हिस्टोन ऑक्टामर फॉर्मेशन हबा और ए जो हिस्टोन ऑक्टामर बनेची ए हिस्टोन ऑक्टामर आगो जाके फॉर्मेशन करिबा कोन हिस्टोन आगो जाके ता तारी परे डीएनए रैप करके हिस्टोन रो फॉर्मेशन हबा न्यूक्लियोसोम तो आगा आसबो डीएनए डीएनए इफ यू सी डीएनए देन डीएनए आगो जीवो बाइंड करिबो का सहित हिस्टोन सहित और डीएनए हिस्टोन सहित बाइंड करले कोन बनै दबो न्यूक्लियोसोम this nucleosome आगो जाएंगे कंडेंस से बने दबो क्रोमैटिन्स then chromatin package we have two options one is euchromatin first then बनेबो आहुरी कंडेंस से कि heterochromatin euchromatin and heterochromatin then that heterochromatin will again get condensed to produce chromatids and these chromatids will further going to produce chromosome clear so this is the entire pathway a bhaliya DNA amar formation है clear Any doubt? Okay. So just take a snapshot or take a screenshot or you can just write it down. ଏଠି ଜିନିଷ ହେଲା ପରେ ଆମେ ପୁଣି ନ୍ୟୁକ୍ଲିୟସ୍ ପାର୍ଟ କୁ ଏଣ୍ଟ୍ରି କରିଯିବା. So nucleus, nucleolus, chromosome, chromatins. Then ଆମେ ପଢିବା ତା ବିଷୟରେ microbodies and we'll wind up the class. I hope till this everything is clear. But still, if you have any doubt, you can ask. So I am erasing it all and we'll move to the next segment. Now, in the nucleolus, one or more non-membranous spherical bodies are there and it is continuous with the nucleoplasm and it is a site for ribosomal RNA synthesis. It is one formation hochi rRNA or ribosomal RNA formation hochi. One or more non-membranous spherical bodies present in the nucleus that is called as nucleolus. It is continuous with the nucleoplasm. Then the cell cytoplasm, so the nucleus and nucleoplasm, out as a continuously present. It is the site for ribosomal RNA synthesis. What is ribosomal RNA? It is basically rRNA, ribosomal RNA. Ribosomal RNA is the structural RNA. Ribosomal RNA is called structural RNA. Why structural RNA? Because it produces ribosome. As it produces now. This is the nuclear envelope. You see two membrane envelope. This is the chromatin which get condensed inside this part. This chromatin thread like structure chromatin ये आगे हो जाएंगे condense होगी ये भड़िया condense होगी chromosome रे convert ହେଇଯିବେ. And ଏଇ जो chromosome बनिबो पुणी एक chromatid ଆଉ ଗୋଟେ chromatid बनेगी sister chromatid formation करिवे and sister chromatid मिसी ଗୋଟେ chromosome बनेबो ए माने आगे हो जाएगी condense होगी chromosome रे formation हेबे. That is the nucleolus, the center part, non-membranous, membrane किसी नहीं, and these are the cisternae present of this endoplasmic reticulum रा cisternae present अछि and that is the nuclear pores. जोटे दीटा का membrane आपस में fuse ହେଇଯାଇଛନ୍ତି and due to that fusion a port like substance is created that is called as nuclear pore. Next, so what is a chromosome? A human cell has about 2-meter long DNA distributed among its 46 or 23 pairs of chromosome, and every chromosome has a primary constriction centromere on the sides on which distinct disc-shaped structure called as heteropores or kinetopores are present. So, okay, so once again, let's understand this very important topic that is a chromosome. So, what is a human cell is having 2-meter long DNA. So, approximately that is 2.2 meter, 2.234 meter long DNA, which is distributed among its 46 number of chromosomes. So, we remember human beings have 46 chromosomes, and that 46 chromosomes are distributed in 23 pairs. 23 pairs. These 23 ରୁ 22 pairs are autosomes and only one pair is allosome. These autosomes are called as somatic cell formation. Allosome are also called as sex chromosome. In case of male, you'll find 22 pairs of autosomes and 22 pairs of autosomes and one pair of X and Y. But in case of females, you'll find 22 pairs plus XX chromosomes. Every chromosome has a primary constriction. All chromosome have a primary constriction type that is called as centromere. That is part is called as centromere. Every chromosome, this part is called as a centromere. This part is called as centromere. जोउठी भी तुम देखिबा गोटे क्रोमोसोम टिको से माने कोन करछ ना आपस में बाइंड होला बेले गोटे प्राइमरी कंस्ट्रक्शन बनाइथि. That primary constriction is called as your centromere. That part is called a centromere. Next, of which a disc-shaped structure is called kinetochore are present as charipote. But a disc-shaped structure is that is called as kinetochore. The centromere charipote, but a disc-shaped structure present is that is called as kinetochore. E the chromosome hela, the centromere hela, a D charipote disc-shaped structure present is that is called as this kinetochore. Yes, send it out. Yeah. Now you can see again that part that is a chromosome which is having a centromere, central is centromere T, and then the centromere charipote ki achi kinetochore proteins man achandi. This is the centromere. Our centromere bhitare man kon achan. This is the chromosome. This is a chromosome. In between this chromosome, chromatin fibers are there. These are the chromatin fibers. They are just highly condensed to produce a chromatid. This single part is called as a chromatid. As you can all see, this is called as chromatid. And a chromatid, ये गोटे chromatid, ये भी गोटे chromatid. And ये जो chromatid माने अच्छे ये chromatid माने आपस में मिसी की लीडा sister chromatid homologous sister chromatid मिसी कौन बने चली गोटे chromosome टे formation कर चली. Then the chromosome formation है this one is a chromatid, this one is also a chromatid, and when two chromatid get condensed with each other at a single circular point, that is called as centromere. Then that is called as your, this part is called as your okay. Now ये जो structure है, if we zoom this part, जब ये part को zoom करके देखियो पिला बने, तलसे हमको किसी ए वाला structure टे show करिबो. कौन show करिबो ना हमरे टे चारिपटे histone octamer अच्छी. This is H2A, this is H2B, this is H3, and this is H4. पूरे ऊपर से या H2A, पूरे ऊपर भी H2B, पछपटे H3, and then पछपटा H4. तो ये मिसी कौन बनेचन गोटे हिस्टोन ऑक्टामर टे बनेचन. तो 1 2 3 4 पछपटे 5 6 7 8 1 2 3 4 5 6 7 8 ए बडा हिस्टोन ऑक्टामर बने अछंती. और ये जो हिस्टोन ऑक्टामर बने अछंती एई माने आगो जाएंगे कौन फॉर्मेशन करछंती ना गोटे न्यूक्लियोसोम फॉर्मेशन करछन. दिस पार्ट इज कॉल्ड एज न्यूक्लियोसोम. Why it is called nucleosome? Because it having a 200 base pair of DNA is getting very slow. 200 base pair of 200 base pair of DNA is getting very slow, dear participants. Two hundred base pair DNA wrapped around a histone octamer. It's getting very, very difficult to write over here. Two hundred base pair of DNA wrapped around a histone octamer to make a that is called as your nucleosome. और जो न्यूक्लियोसोम अच्छी न्यूक्लियोसोम आखिरी कंडेंस हे बनेबे क्रोमैटिन क्रोमैटिन पुनी कंडेंस हे बनेबे क्रोमैटिड और क्रोमैटिड कंडेंस हे बनेबे क्रोमोसोम दैट इज ऑल अबाउट क्रोमैटिन क्रोमोसोम एंड जो हिस्टोन ऑक्टामर माने अछंती मधुरे जो तुम दिस पार्ट देखो जो दिस पार्ट इज कॉल्ड एज जो लिंकर डीएनए दैट पार्ट इज कॉल्ड एज that part is called as linker DNA. That is called a linker DNA and जो linker DNA अछंती ए मतलब लिंक करके रखुचंदी आ सहित H1 हिस्टोन प्रोटीन भी थाए सहित हिस्टोन now you can see very well in this diagram that is a chromosome is here chromosome is a 1400 nanometer diameter ଅଛି कि तुम्हारा क्रोमोसोम क्रोमोसोम भीतर जो सिंगल क्रोमैटिड अछि that is 700 nanometer क्रोमैटिड टी then से क्रोमैटिड जब अबे तो डीकंडेंस हबो then ता भीतर 300 nanometer हेटेरोक्रोमैटिन आसीबो then ता भीतर 30 nanometer यूक्रोमैटिन आसीबो then ता भीतर 10 nanometer न्यूक्लियोसोम आसीबो और then ता भीतर 2 nanometer डीएनए आसीबो तो तबे अब रिवर्स प्रोसेस से भी देखि परिबो like in the reverse process you can see like first we are having a 2 nanometer length DNA is having जो 2 nanometer length DNA अछि से DNA आगो जाइकी न्यूक्लियोसोम चारिपटे रैप करिवो न्यूक्लियोसोम चारिपटे कोन हिजो हमर जो हिस्टोन ऑक्टामर चारिपटे रैप करिवो and जेबे गोटे 2 nanometer width DNA टी गोटे histone octamer which is having 8 number of histone protein that is H1 that is H2A H2B H3 and H4 two molecules of H2A two molecules of H2B two molecules of H3 and two molecules of H4 the चार्टा रो नेकी जो ऑक्टामर फॉर्मेशन हिची से ऑक्टामर चारिपट से रैप करिवो देन सेटु फॉर्मेशन हिची कोन गोटे न्यूक्लियोसोम then these nucleosomes are coiled in between तांको भितर जो लिंक करके रखि that is called as the linker DNA or the linker protein that is again nothing but H1 histone protein and then say nucleosome आहुरी condense formation करिवो euchromatin and say euchromatin आहुरी condense formation करिवो heterochromatin and say heterochromatin रो formation है जो chromatid and then finally the chromatid will make into chromosome that is the all scenario first we'll having DNA first we are having DNA अब प्रथम DNA आसीबो DNA आसला पर DNA को हमें रैप करिबा first we are having DNA we are having DNA DNA कितना नैनोमीटर थिला पिला माने 2 नैनोमीटर काइंडली बेयर विथ मी ये बहुत टेक्निकली प्रॉब्लम आसुचे लिखला बडको Sorry, if ପିଲାମାନେ ହଉନି କିଛି ଏପଟେ problem ହଉଛି ତ you can see this is the 2 nanometer DNA ये जो DNA present ଅଛି ଯେ DNA condense ହେଇ ବନେଇବ ଏଠି histone octamer ଆଉ ଏ ଯୋଉ histone octamer formation କରିଛି ସେ histone octamer ଆଗକୁ ଯାଇକି formation କରିବ କଣ nucleosome ଆଉ ଏ nucleosome ଆଗକୁ ଯାଇ condense ହେଇ ବନେଇବ ତମର ଏଠି କଣ euchromatin ଆଉ ସେ euchromatin ଯାଇକି ଆହୁରି condense ହେଇ ବନେଇବ heterochromatin ଆଉ heterochromatin ଯାଇ condense ହେଇ ବନେଇବ chromatid formation କରିଦବ chromatid and chromatid आगो जाए formation कर देगा chromosome. This is a whole sentence. This is chromatid. This is chromosome. So this is your centromere. This part is your centromere. And a part चारिपटे गोटे kinetochore protein. A part चारिपटे kinetochore protein टे formation हवा. I hope you understood. The participants, if you have any doubt, you can ask me in the chat box. Now, based on the position of centromere, chromosomes are of 4 types. So, chromosome जो होती centromere ର position को नेकी चारि प्रकार भाग देई परिबो कोन अछि सबट प्रथम जो देखिबो centromere मझिरे अछि it is called as but जो मझिरे अछि अबिका part जो हमर chromosome ता मझिरे that is called as metacentric तो टिके ऊपर को अछि सब metacentric आउ टिके ऊपर गला acrocentric total ऊपर पड़ेला से हेगो telocentric so on the based on the position of centromere chromosomes are of 4 types. How many types chromosomes are there? Chromosomes are of 4 types. And what are those 4 types chromosomes? Type of chromosome on the based on the position of centromere, position को base करके based on the position of centromere, our chromosome 4 प्रकार रहिले. One is a metacentric, second is submetacentric, third is acrocentric, and fourth one is a telocentric chromosome. So metacentric, this is a metacentric chromosome. Metacentric means the pate equal structure present achi. This is called as a metacentric chromosome. If a metacentric tikke upuru paleba bika, that is called as one short arm and another long arm, that is called as submetacentric. If we further move ahead, then we got acrocentric chromosome, one extremely small and one extremely large. And if you move further ahead, then it is called as telocentric. This part is called as the short arm, this one and this one. And then this part is called as a long arm. This entire part is called as a long arm. And this part is called as a short arm. And this part is called as a centromere, which is present in the middle constriction. And remember, pilamane, one chromosome always contains one centromere. गोटे chromosome पाखरे गोटे ही centromere थिलो. अब centromere position को लेके हम जब divide करते, it can be metacentric if the centromere is present in the middle, dividing the chromatids into two equal halves, or it can submetacentric when the centromere is little bit away from the center and it is towards a periphery. It is called as submetacentric. Then part होती के further आगे गली it is called as ectocentric and पूरा extremely proximal to पड़ेगा end to पड़ेगा then that is called as telocentric chromosome. Centromere is very close to its end, forming one very short and one very large. One very short and very long arm. Telocentric means terminal centromere. Centromere is present at terminal end. पूरा end रखियो. So any doubt you wanna ask? The participants, Vishwajit Vicky 625460, Danta Higher Secondary School, Parjan Higher Secondary School, Sahil, Sukruli Higher Secondary School. Any doubt, dear participants? Then we come to the microbodies, that is the crystalline core of the structure. These are membrane bound minute vesicles that contain various enzymes present in both plant and in case of animals. ये plant मान पाखे भी थाए, ये animal मान पाखे भी थाए, और ना हेगला कौन microbodies. If you take example of one microbodies, then plant पाखे गोटे microbodies टी अच्छी, animal पाखे भी से microbodies टी अच्छी, ता ना हेगला कौन peroxisome. Peroxisome. It basically helps in C2 cycle. C2 cycle also called as photorespiration. This is called as photorespiration. Like कितने slowly लिखा हो जाएगा. Peroxisome is also called as C2 cycle. Peroxisome रे हेते हैं, and that is called as photorespiration. And if photorespiration है कौन हेते हैं पिला माने हमारा कार्बन डाइऑक्साइड जगह ऑक्सीजन टा फिक्स हो जाता है फोटोसिंथेसिस रे, and सेटू कौन करता है ग्लाइकोसिलेट साइकिल चलता है. और से ग्लाइकोसिलेट साइकिल रो हमारा जो ग्लाइसिन सेरीन ये सब अमीनो एसिड फंक्शन होता है. तो peroxisome is a kind of microbody which is present in case of plant and animal. In case of plant, it does the glycolate cycle, and there it gets reduced into photorespiration and finally produces two molecules of 3-phosphoglyceric acid, which enters into the Kelvin cycle. So these are basically membrane bound minute. Minute means small. Now, the difference between plant cell and animal cell, as you can all see, plant cell, cell wall is present, animal cell, cell wall is absent. In plant cell, plastids are present. In animal cells, plastids are absent. In plant cell, a large central vacuole is present. In animal cell, many small vacuoles are present. While vacuoles are basically absent, in plant cell, centrioles are absent. Animal cell, centriole is present. तो टेक्निकली प्लांट आउ एनिमल को सब डिवाइड करबो चारटा सी रे काम हो जाय छै 4C लिखित होउनी किसी को भी जिंस कोन करबो एठी तो फर्स्ट सी रे हे गला सेल वाल प्लांट पाखरे अछि एनिमल पाखरे नाही तापर आसी गला सेल रो सेंट्रल वैक्यूल सेंट्रल वैक्यूल अछि सेंट्रल वैक्यूल नाही तापर पड़ा सो क्लोरोप्लास्ट प्लांट पाखरे अछि एनिमल पाखरे नाही तापर पड़ा सला सेंट्रियोल प्लांट पाखरे नाही एनिमल पाखरे अछि जदि डाउट हला पचरे कि So basically the four different parts. I hope तुम जाई बुझी पारथिबो. The difference between prokaryotic cell and eukaryotic cell. Prokaryotic cell. ये बहुत सारे ये स्लो होची लिखला आलो किछि भी कला आलो प्रोkaryotic करियो एपडे लिखला हां सेपडे भी लिखला आलो भी अच्छा एतो थोड़ा वाइंड अप कर दियो 5-10 मिनट. Now, the difference between prokaryotic cell and eukaryotic cell, if you observe, prokaryotic cell generally smaller, prokaryotic cell are generally smaller in size, the eukaryotic cell are generally larger than the. The difference between prokaryotic cell and eukaryotic cell is like this. Prokaryotic cell are generally smaller, while the eukaryotic cell are generally larger. Then the genetic material in the form of nucleoid, here you won't get the nucleus, you are getting nucleoid, but here genetic material is present in the form of nucleus. In nucleus form and in nucleoid form. What's the difference between nucleus and nucleoid? The nucleus चारिपटे तार membrane present अछि, nuclear membrane nuclear envelope present अछि, and nucleoid चारिपटे से membrane present नहीं. Nuclear membrane is basically absent. Nuclear membrane कौन अच्छी नहीं आप absent अच्छी और nuclear membrane टी आप present अच्छी. The membrane bound organelles are absent. They don't have any kind of membrane bound cell organelle. आपका basically absent है. But in case of eukaryotic cell, the membrane bound cell organelle is basically present. Circular DNA. The DNA is basically circular in nature. As it's a prokaryotic cell, but here the DNA is linear in structure. Our linear money like this, double helical linear structure through DNA. DNA भी एठी इक्वल वैल्यू होनी चाहिए. This is double stranded linear DNA and apo DNA के अंदर double stranded circular DNA. Double stranded circular DNA. Then ribosomes are of 70S type. Ribosome आगे पाखे 70S type पर थांती and ribosome आगे पाखे थांती both 70S as well as 80S. 70S भी 80S भी. One more thing, in case of prokaryotic, P रहेगा plasmid. They are having plasmid. And what is plasmid? Extra chromosomal circular double stranded DNA. That is called as plasmid. They have this plasmid, whereas they do not have that plasmid. त्यामध्ये plasmid टी नाही. Now, time to get some question answer. So, dear participants, I am requesting all of you to kindly participate and answer these questions so that we can check your understanding level. The first question, the nucleus was first discovered by whom? Who discovered the nucleus? The nucleus was first discovered by, your options are Robert Hooke, Robert Brown, Leeuwenhoek, and Purkinje. I sincerely request all the participants to participate and answer this question. The nucleus was first discovered by whom? Robert Hooke, Robert Brown, Anton von Leeuwenhoek, or Purkinje? Yes, who wanna tell this answer? It's a very basic question, very easy question. Sukruli Higher Secondary School, Mayurbhanj, or Janta Higher Secondary School, Dhenkanal, or the other participants who are watching this class right now, I'm requesting all of you to kindly participate, kindly give the answer in the chat box, or you can unmute yourself and tell me the answer directly. Some very basic questions are here. The nucleus was discovered by, the name of the scientist is Robert Brown. The option B is correct, that is called as Robert Brown. The outer membrane of the nuclear envelope is usually continuous with the dash. The nuclear envelope जो data membrane थिला, गोटे inner membrane, गोटे outer membrane. That outer membrane of the nuclear envelope is usually continue करता है. कहाँ तरह continue करता है? Whether it is the Golgi apparatus, or it is the endoplasmic reticulum, or it is the lysosome, or it is the peroxisome. So what will be the right answer? The right answer is endoplasmic reticulum. The right answer is endoplasmic reticulum, option number B. The non-membranous granular structure present inside the nucleus, which is a site for rRNA synthesis and ribosome assembly, that is called as dash. Question बहुत बढ़ा दीजिए. The non-membranous granular structure, जो structure है कौन membrane नहीं, जो structure किसी membrane नहीं और which is present inside the nucleus, and its function is also given, that is it helps in rRNA synthesis and ribosome assembly, then the structure is called as nucleolus. Option B, correct answer is nucleolus. The genetic material in prokaryote is called as dash. ଏବେ ବି recently ପଢିଛେ prokaryote ର genetic material ନା କଣ, whether it is nucleus or nucleolus or nucleoid or chromatin, and the right answer goes to option number C, that is called as nucleoid. The fundamental unit of chromatin packaging consisting of DNA wound around the core of 8 histone protein. That core is called as dash. The fundamental unit is called as dash. Whether it is solenoid or it is chromomere or it is nucleosome or it is chromatin fiber. And the correct answer here is nucleosome. Which of the following is not a part of a nucleosome core? Nucleosome core रो किए part नुहे H1 histone, H2A histone, H3 histone, H4 histone. And again, the correct answer is H1 histone, because in the nucleosome we are having a protein octamer, that is H2A, H2B, H3, H4, and H1 is called as linker. It is not a part of the nucleosome core. During which stage of the cell cycle are chromosomes best visible under light microscope? The light microscope ରେ ସବୁଠୁ ଭଲ chromosome କୋଉ time ରେ ଦେଖାଯାଏ? Whether it is a interphase or the prophase or metaphase or anaphase. ତ chromosome କୁ ଦେଖିବାର ଅଛି, ତାହେଲେ metaphase ର end, anaphase ର start, ସେଇ time ରେ clearly ଦେଖିପାରିବ। ତ of course anaphase ବେଳେ chromosome split କରିବାକୁ start କରିଥାଏ, but metaphase time ରେ ତମେ chromosome କୁ आराम से देखिପାରିବ। A metaphase chromosome consists of two identical strands, and these two identical strands are called as dash. The answer is chromatids or centromere or telomere or kinetochore. The correct answer is chromatids. Clear? The question 8टा का हम आंसर पाइगले। सब question answer पाइला। First question answer is Mr. Robert Brown. Question number 2 answer हमर कौन आसिला? The endoplasmic reticulum. Endoplasmic reticulum. Question number 3 answer हमें कौन पाइले? The answer is nucleoid. Sorry, non-membranous structure T. No, it's not nucleoid. हमें answer पाइले टी कौन? The non-membranous structure that is called as the nucleosome. Sorry, nucleolus. Next, genetic material prokaryotes has genetic material ना कौन है क्या nucleoid. Then fundamental unit of chromatin packaging is called as nucleosome. Which of the following is not a part? That is a H1 histone. During which stage of cell cycle chromosome best visible? Answer is metaphase, late metaphase, and metaphase chromosome जो हमें divide ହେଇକି बनेଇଥାନ୍ତି, that is called as chromatids. Clear? Question 8 का answer तुम screen पे रहला. Take a screenshot or whatever you want to like. You can join it from here and you can take. Now, we'll move to the last part of this chapter. The point on the chromosome where two sister chromatids are held together. Two sister chromatids are held together. The point on the chromosome where the two sister chromatids are held together, that is called as your centromere. The terminal non-coding repetitive DNA sequences on chromosomes that prevent erosion of genetic information are called as dash. The terminal non-coding repetitive sequences, that is called as your telomeres. The structure on the centromere where spindle fibers are attached during cell division. Spindle fibers are attached to the kinetochore attached. Based on the position of the centromere, a chromosome with arms of roughly equal length. Equal length, the answer is metacentric. What is the composition of chromatin? Chromatin ne ki pila chindi? Chromatin contains DNA and histone protein. DNA सहित histone protein. Which type of chromatin is transcriptionally active and less condensed? Transcriptionally active जब देखिबो our answer हैला U chromatin. Transcriptionally active and it is less condensed. The beads on string structure in chromatin is formed by beads on string structure is basically bonds nucleosomes connected by linker DNA. Nucleosomes connected by the linker DNA. Then which of the following statement is about the nuclear pore complex is incorrect? It allows free transport of all molecules. It regulates the passage of molecules. It is formed by the fusion of two nuclear membrane and large molecules require active transport. Option B correct achi. Option D correct achi. Option C correct achi. The right answer है option A. It allows free transport of all molecules. समस्तों से freely छाड़े नहीं. Hereby we have completed this entire chapter. Thank you so much for your kind attention and we'll definitely meet again.